Hello, everyone. Thank you for joining us today. This is our first annual virtual pitch day that we're having with our companies. We really appreciate everyone taking the time to be here. This should be a great opportunity to get to see some fantastic early stage companies. I'd also like to thank my team for working so hard to put this together. It's going to be a great day for all of us, and I look forward to hearing all of the pitches myself. What we're going to be doing is having everybody do 10-minute teaser pitches. There'll be 5 minutes of Q&A after that, and then we'll quickly move on to the next company. It's our goal to keep everything on schedule, so we will try to push people along and when they're done with their pitch. So with that in mind, we will get started from here. The first company up will be Exio, and our speaker today is Joseph Carroll. I'll have him introduce himself and we'll get started from here. Thanks again, everyone.
Hi, everybody. I'm Joseph Carroll. Do you all hear me? Hi, everybody. I'm Joseph Carroll. I'm the CEO of Exio Biosciences. I'm going to share my presentation now. Michael, is that coming up for you? Yeah, okay. All right, I'm happy to talk about Exio Biosciences today. I am the acting CEO, and there are a few logos on this slide, so I'll take you through the logos one at a time by way of some context. I am in the life science group at IP Group. IP Group is an early stage strategic investor, and we play a very heavy operational role in the early stage companies. This is why I'm acting CEO. We're giving about a third of the pitches today, so you'll hear from a number of my colleagues who are acting CEOs for some of the other companies. Second logo here is UPenn. I don't have to tell you about UPenn, very venerable and innovative institution, but I will tell you that we leverage the UPenn ecosystem pretty heavily. And our co-founders for Exio Biosciences at UPenn are Professors Wei Guo and George Xu. And we also leverage other aspects of UPenn. So our logo here for Exio is designed in the PCI Ventures Penn Design Practicum program by Rachel Liu. So we're very thankful for that. All right, Exio Biosciences is really playing in the immunotherapy space. And you'd have to be living in a cave to not know about the disruptive innovation that's been brought about by inhibiting a single pathway, the PD-1, PD-L1 pathway, a way of cancer cells has of modulating the immune system. There are antibodies these days, you know, these PD-1, PD-L1 antibody therapeutics for cancer are really innovative and disruptive. They're very effective, but only in a subset of cancer patients. So hence the need for a better diagnostic. Some of the key clinical questions here that one can ask if you're a clinician that are really critical is, will my patient respond to PD-1 therapy? And if they're not responding to the monotherapy, is a combination needed? And then once they're on therapy, is the patient responding to therapy? So Exio Biosciences has a blood-based diagnostic test to address these very questions. This test is much more specific and sensitive than existing diagnostics. Today, we're currently closing a $1.2 million seed round. We're going to be doing a Series A in mid-2022, where we'll be building a syndicate. I'll talk about at the end to do that. All right, so the immuno-oncology diagnostics market is really driven by the massive immuno-oncology therapeutics market. Currently, the market for agents targeting PD-1 and PD-L1 is about $16 billion. It's expected to grow to more than $70 billion by 2026. It's actually experiencing accelerated growth right now. And the reason that the growth is accelerating is because of the large number of clinical trials involving this pathway. As of September, there are more than 4,000 active clinical trials involving PD-1 and different combinations in the clinic for cancer patients. So these trials are really presenting a massive market opportunity, both for the therapeutics market, as I stated earlier, but also the diagnostics market. So the immuno-oncology diagnostics market is currently about $3 billion. It's going to double by 2024. The fastest growing segment of this market is the companion diagnostic testing. And this is the arena that Exio will be moving into. Now, even though the current diagnostics tests are very poor, they're still used by a vast majority of the clinicians. So about 90% of the time, clinicians use a diagnostic right now in lung cancer, and about 75% of the time in metastatic melanoma, clinicians use an existing test. So there's a compelling case to be made for better immuno-oncology diagnostics. As I said earlier, the checkpoint inhibitors currently to PD-1 and PD-L1 are very effective, but only in a subset of patients. In fact, in metastatic melanoma, only about 30% of the patients have a durable complete response. And in lung cancer, it's even worse. It's about 20% of the patients. Now, you might think, well, you can just have a combination if the agents individually aren't working well. But it turns out that there are significant adverse events as you start to combine these immuno-oncology agents. So you got to be really careful about guiding therapy. Of course, the cost is also more significant as you come up with combinations of treatment. So for all of these reasons, clinicians want to be able to predict PD-1 response better than they can currently do using current methods. And ideally, they would like to have a liquid biopsy that can really help them predict response to PD-1 therapy, especially for the combinations. So this brings us to Exio Biosciences. Exio's current prototype test is based on this really landmark publication by our co-founder Wei Guo in Nature in 2018. And what Wei showed in that paper was, although cancer cells often turn off T cells and other immune cells in the tumor itself, and this is where most of the focus is right now in the tumor, it turns out that cancer cells also secrete exosomes with PD-L1 on the surface. And these exosomes are then able to inhibit T cells in the periphery. So Exio is basically intercepting this signal and using these PD-L1 exosomes as a way to interpret what's going on in the tumor. So to summarize the biology here, melanoma cells secrete exosomes with PD-L1. These secreted exosomal PD-L1 vesicles can actively suppress the CD8 T cell immune response in the circulation. And patients have variable levels of these exosomal PD-L1 molecules that correlate with response to therapy. And that's pretty critical. Now, I'm only talking about PD-L1, but in addition to PD-L1, there are, of course, numerous other checkpoint inhibitors. And Wei is now characterizing these other checkpoint inhibitors on exosomes. And that's helping Exio to build a test menu. So I said earlier that there are other diagnostics that are currently being used. But I'm going to tell you first about the prototype assay that Exio has. So ideally, you want a very specific and sensitive assay that is both predictive and prognostic to guide therapy. And that's exactly what Exio has in this prototypic assay that I'll show you. So you gauge assay performance by looking at sensitivity versus specificity. And when you do this in a retrospective clinical data set, as we did for about 20 melanoma patients, 40 patients in total, some half responders, half non-responders, and you correlate the test for sensitivity and specificity, the existing PD-L1 assay that is often used has an AUC of about 0.7 here. So this is just looking at total PD-L1 free in the blood. However, when you look at exosomal-based PD-L1 and do the assay for exosomes, you're able to raise the AUC to 0.9. So this is a really sensitive test that's much better than existing immuno-oncology diagnostics. The existing immuno-oncology diagnostics, even though it's currently a $3 billion market, they're largely based on genomic evaluation. Companies like Foundation Medicine are looking at tumor mutational load. Other companies are looking at T cell location or T cell receptor repertoires. Roche Ventana has an approved companion diagnostic test for PD-L1 immunohistochemically, and still other companies like NanoString and HalioDx are looking at tumor inflammation guided by RNA. The challenge with these tests is that none of them are liquid biopsy based. They're all biopsies of the tumor themselves, and that comes with a load of challenges. Additionally, none of these tests have an AUC that's greater than 0.7. And even in our prototypic assay, we already are at 0.9. So for all of these reasons, Exio is undergoing a very ambitious roadmap to bring this exosomal PD-L1 assay into a CLIA lab in the next several months. So we aim to be in a CLIA CRO by July of this year, initially with a test for metastatic melanoma. We'll be moving into other solid tumors like lung cancer and triple-negative breast cancer in early 2022. As I alluded to, in addition to exosomal PD-L1, we're looking at other checkpoint inhibitors. So we already have some R&D going on for these molecules. We'll bring additional tests online in early 2022 into a CLIA lab that give Exio a really compelling test menu for immuno-oncology diagnostics. Additionally, then, as we move to the Series A funding in 2022, we will be looking at a roadmap to develop our exosomal tests as companion diagnostics or LDTs. And then we'll use the Series A funding to bring those through regulatory filings. Along the way, we'll be doing biopharma deals with our assay platform, of course. Okay, so in summary, what I've shown you is that Exio Biosciences has a truly disruptive diagnostic for immuno-oncology therapeutic applications. We have some major technical and commercial milestones that we'll be achieving hopefully in the next year. And our fundraising goals, we're currently, as I said, closing the seed round and using those funds to bring the assays into a CLIA environment. We anticipate that in July 2022, but could be earlier if we're able to build a syndicate earlier, we will use these funds to bring the companion diagnostic through development. I want to give special thanks to Bhavana Mohanraj, who is in PCI Ventures, also an Exio board member, as well as John Todd, who is our most important advisor and was a former CSO at Singulex. Okay, with that, I think I'm at time, so I'm happy to take a couple of questions.
Great. Thank you so much, Joseph. That was a fantastic pitch. If anybody has any questions, we have just a few minutes here. There we go. So we have a question, Joseph, about your exit strategy.
Yeah, always important to think about. We imagine that as we, after our Series A funding, as we have a companion diagnostic test that is undergoing final filings to be used in the clinic, we imagine will be acquired at that point. So that's our exit strategy, is really to be acquired, say 2023, 2024, I think is when we'll be, building enough value for that to be possible.
Okay. Do you have an assay pilot in progress?
Yeah, we have a prototypic assay. We're just transferring it now to the CRO. So we're undergoing some validation studies soon to make that assay happen.
And talk about your IP protection.
Yeah, we have really good IP protection. Wei has been great about protecting this in advance of his Nature paper. So we have 2 filings, one of which is published and is covering both methods of exosomal PD-L1 and other checkpoint inhibitors in a variety of solid tumors.
Excellent. Thank you so much, Joseph. I appreciate it. And if anybody has an interest in learning more about Exio, please reach out to Joseph separately. Thanks so much.
Time, everybody. Thanks so much. Take care.
And we will be moving on to our next company is Chip Diagnostics. It'll be Nena Golubovic will be presenting, and she's getting her screen reader. This is a second IP group company. So they were the initial investors, helped us get the company started. And I will hand it off to Nena. Thanks so much.
Can you hear me? I assume you, okay, very good. Well, so thank you, everybody. Thank you, UPenn, for this opportunity. My name is Nena Golubovic, and I'm here on behalf of Chip Diagnostics today in the role of CEO. I'll talk about our novel biomarker discovery and diagnostics platform. It's a short time, big story to tell. So my goal is really just to paint some really broad strokes around the areas that I think are very important. That is to address kind of state of readiness of our technology. How do we think about commercializing our products and solutions? And then a little bit about the ongoing fundraise. It's going to be somewhat scripted because of the time limitations, but we'll be very happy to have more discussions on any subjects of interest. Just make sure I'm in control of my screen. So Chip Diagnostics was founded in 2015 by Professor Isador, UPenn and IP Group. The company currently has 2 full-time employees, experienced leadership and advisory team. Chip DX technology development has been substantially funded to date through academic collaborations as well as private funding from IP Group, and resulted in so far 8 peer-reviewed publications, 4 patents issued and pending, and most recently in commercial collaborations and sponsorships. Our mission is simple, but also very big. We want to exponentially improve healthcare outcomes by developing tools to aid clinical decisions by enabling organ-specific information. This is where we are unique from liquid biopsy samples. Just a little bit background on liquid biopsy. I'm sure everybody knows very well, so I won't go deep there, but blood-based biopsies enable access to genetic material from tumor without the need for invasive procedures. And technological advances have generated lots of interest in this field, with lab-developed tests for liquid biopsy in some cancers already available. So this is all very promising, but often with limitations, largely due to biological diversity and heterogeneous nature of the disease, which combined often result in limited actionable insight with current technologies. So at Chip Diagnostics, we see that there is an important and complementary role that exosomes could play in liquid biopsy, sitting adjacent to genomics and other tools and technologies. So a little bit background on exosomes. They're the smallest type of extracellular vesicles that are found in circulation. They contain a complex cargo of content, and they are derived from the original cell that they come from. So they contribute to a spectrum of biological processes in both health and disease. And because their contents are molecular signatures of the originating cells, they're being evaluated in an array of potential diagnostic and therapeutic applications. At Chip Diagnostics, we look at exosomes as the cell's remote sensors in a way. If they can be isolated for analysis, they represent a powerful data source for insights that are not otherwise available. So there are, of course, challenges. One of the key challenges is the ability to isolate exosomes in order for this to become a translational tool. As an example, ultracentrifugation is, I think, a gold standard at this point, comes with some issues around yield, purity. It's a labor-intensive technique and requires large samples, it's time-consuming, etc. So there is clearly a need for translational tools to aid clinical decisions, for which is where Chip Diagnostics comes in. So this is maybe a good time to perhaps introduce Exotempo technology platform. It's really a combination of hardware and then informatics piece, which so basically. What's unique about our hardware, Professor Isador and his team have invented what I like to refer as probably one of the most elegant and nanofluidic sensor systems, which rapidly scans each individual 1 of trillion exosomes in a clinically relevant sample. So this is very difficult to do, and what enables it is a kind of a unique nano sensor architecture, which is enabled by sophisticated engineering, very much borrowed from the microelectronics industry, and that combined with the fundamental principles of magnetic force and flow physics. So when in comparison with some competitive bulk magnetic or other technologies, I would highlight that ExoTempo is an orders of magnitude different approach in that it screens each individual target exosomes against a massive background, which is typically present in a clinical sample. So from the standpoint of scale and microfluidics, this is a massive technological challenge, and so is chip diagnostics' accomplishment in the ability to solve that problem. So in addition to our proprietary exosome isolation technique, chip, as I said earlier, is differentiated by its machine learning approach to analyzing the captured exosomal content. So to correlate multiple exosomal biomarkers, chip applies a supervised machine learning technology to our proprietary data sets, and that's the whole another level of how this technology adds value. In terms of validation, I will say that the clinical significance and value of the new insights enabled by ExoTempo have been demonstrated in multiple clinical studies across multiple patients, across multiple diseases. CHIP team has demonstrated ability to detect disease early, to differentiate between patients who are good candidates for surgery versus those who are not, and is currently evaluating ExoTempo in identifying patients who will respond to an IO therapy versus not. So this systematic progress has resulted in multiple peer-reviewed publications and is driving strong traction and interest from commercial collaborators, the likes of Johnson & Johnson, United Health Group, and LabCorp. I will say that CHIP plans near term to leverage these collaborations to scale the sales of our first product configuration, which is an instrument cartridge system for enrichment of tissue-specific exosome subpopulations. Through these collaborations, CHIP will also gain access to additional clinical samples and disease expertise for further technology validation. We are, as I said, at the early commercialization stage. Over the next 12 to 18 months, our plan will not only result in product revenues, but also a larger and broader validation of ExoTempo by third-party users. We believe this will drive adoption and scale up of our business, but it will also drive the company to a significant value inflection within the next 12 to 18 months. In terms of our corporate development roadmap, I will say that despite COVID, 2020 has been a good year for chip diagnostics. The company has its first $350,000 in revenue from orders, first our initial customers, and we plan to scale these revenues in 2021 while leveraging our fully established third-party vendor supply chain. I will highlight that the technology is completely transferred out of the lab. And so all our products, instruments, and disposable cartridges are manufactured by qualified vendors. We feel timing is good. In the market, there remains a need for rapid, sensitive, and cost-effective gold standard method for exosomes. By entering tools market, CHIP will have only scratched the surface in terms of value that our technology potentially unlocks for diagnostic and other use cases and translation into clinical settings. We are fully aware of that, but that said also we feel that by establishing RUO and through clinical validation, CHIP will continue to position for larger scale impact with our solutions and expertise in exosomes while de-risking our technology and business model in capital efficient manner. You will see some details here in terms of how we project our financing rounds. We are aligning that with value inflection points and all of the usual stuff. In terms of the current financing, IP Group is the only institutional investor to date. We are currently raising large seeds, mini A round, whatever one wants to call it. It will very much depend also on who our potential lead investors are at this stage. But basically raising that round to establish the core functionality, to launch RUO business outside of the initial UPenn customers and to continue validation of the technology, specifically in the lung cancer diagnostics and therapy. We have an open convertible with capacity to receive additional funds. and we are in discussions with a couple of lead investor prospects. In terms of the team, as I mentioned before, we have 2 full-time employees who are operating out of Penn Innovation Center. We have a couple of very experienced advisors to the company who are adding value at this point in consulting roles. Some of these roles may be transitioned into the company in a very near term. In terms of our board of directors, a lot of experience there. Our very own Michael Poisel of UPenn, Heiner Dreisman, former CEO of Roche Molecular. We have Guido Batchelor, former CEO of Singulex, Thomas Warto, serial entrepreneur in biotech space. A lot of good expertise, a lot of understanding of industry, but all the way from tools to diagnostics. And good networks. On the collaborators and clinical advisor side, we have several UPenn based, and outside of UPenn, we have Duncan Whitney, who is J&J's head of lung cancer initiative in Boston, and additional kind of advisors, as well as access to the resources. So perhaps I'll stop here because I believe I wasn't watching carefully the time, but maybe I'll just open for questions.
Yes, we're at the time limit, so we will. And thank you for that presentation. That was great, Nana. So we will now look down the list for some questions. So how much of the funding that you're raising is going toward testing the system versus growing the company? And also, what percentage of the product is lost through failures at test?
Sorry, say that again, the second question.
What percent is lost through failures at test?
Well, okay. So the first question had to do kind of a growth. I would say we feel at this point technologies has been so robustly validated and through the process of transferring it out of the lab that this is mostly execution risk. So we are currently focused on recruiting expanding the team basically. So transitioning the CEO role because the company is ready for that. And then also transitioning, onboarding some additional team members. So I would say 60% would go in the next 12 months towards that. The rest of it is pretty straightforward at this point. We will operate out of supply chain we have in place for the time being. And then most of the validation, additional validation kind of goes towards the demonstrating the technology in diagnostic applications rather than the tool space, right? So I would say that's where the mostly risk shifts on the technology side at this point.
Okay, and I also want to mention we have the founder, David Isidore, available as well. So if anyone has questions for him, he's also on the line. And we have another question. Are you still accepting angels and safes?
We possibly could. The whole purpose of this safe right now is really to give company enough time to bring the right syndicate together. Because as you saw in some of my projections, we are pretty ambitious in how we see this opportunity and how it needs to scale. So I would say angels right now are welcome. But I feel that we are quite focused at this point on putting together that equity round. And looking for a lead investor.
And your exit plans?
Favorite question. I think the company will at some point rather soon, maybe within the next 2 years, split. We think this is the unique opportunity where there is a path from RUO to LDT to IVD. You can't do all of that in one organization, so we see that there is that. So that's the first part. In terms of exit, I would say on the diagnostic side, that will be different. It's more of an M&A activity when we demonstrate, for example, diagnostic performance in lung cancer. That would be a good timing to think about. On the RUO side, if exosomes are going to be as big as we think they are, and the tools become so important, I would see that more an IPO path.
Okay. And the last question, what's the gross margin today on your diagnostic assay?
So we still haven't implemented value-based pricing on any such thing. We are really just selling these. It's a pretty good margin because we can, there is a lot of value in our solutions. I would say 60% margin right now we are looking at. Long term, I think that is going to be more of a value-based pricing. So there may be academic versus commercial rates, and depending on where in the value chain we fit, the pricing will be impacted by that.
Great. Thank you so much for that presentation on CHIP. If you have further questions, please reach out to Nena directly or to anybody on the PCI Ventures team. We will get the questions answered and to the right people if we don't have the answer. Thank you, David, as well, for being on the line. And next, we will be jumping to Jerry Ann Thatcher, who is the CEO of FlowBio, and she will be presenting that company next. There's Jerry Ann.
Good afternoon. So I have that, can I share screen?
Yes, you should be able to.
Okay, there we go. All right. Fantastic. Well, good. Thank you, Michael. I appreciate it. Good afternoon, everyone. I'm pleased to be able to introduce you to FlowBio. My name is Jerry Ann Thatcher, and I'm the CEO of the company. And our mission is really to set a new standard for diagnosing bleeding and thrombotic risk. And we understand that emergency departments are chaotic. They're high-pressure environments that require rapid information to ensure timely and cost-effective treatment decisions. So just imagine a patient is rushed into the emergency department, and the triage team and the surgeon all converge rapidly to assess and determine next steps for this patient. And they particularly want to know if surgery is required. And within minutes, they can take vital signs and order a multitude of tests and procedures, which can directly inform treatment decisions. Yet, there is a critical piece of information missing when it comes to bleeding risk and understanding the potential for hemorrhaging to death. And this blind spot takes a significant toll on physicians and their ability to manage very complex and very time-sensitive situations. And FlowBio is designing a tool to remove this blind spot. Our solution is an innovative patent-pending microfluidic platform that is uniquely designed to enable blood flow, and it recreates the blood clotting process on the device. And the benefit of this is that it is allowing for sensitive and accurate detection of the underlying cellular components of the blood clotting problem. So we're really providing clinicians with rapid, actionable information that can drive cost-effective treatment decisions as well as monitoring solutions. And our goal, obviously, is to reduce the downstream complications. And we have developed a working tabletop prototype system that fits into the existing clinical workflow. A nurse or a med tech can draw blood from the patient's central line. A small amount, 5 drops, is transferred to the disposable test cartridge. And when run, we have optical fluorescent images of the blood clotting components that are captured in real time. And our proprietary software then analyzes the data, leading to actionable output of the clotting defect. And it does so within 10 minutes. And with our first test, we'll be able to identify the level of DOACs in the patient's system. And so by providing a diagnostic tool that can really remove this blind spot, we're allowing clinicians to make evidence-based decisions and to really reduce downstream complications and to improve outcomes. And I think it's important to note that this problem or blind spot is not insignificant. It really complicates decision-making for these clinicians. And this is especially true for the 45 million patients who are injured and enter the ED every year, as well as 6 million patients that are on newer blood thinners or DOACs, and we know them as Xarelto, Eliquis, and Pradaxa. And keep in mind that 25% of injury patients that come into the ER actually have a clotting defect, but it often goes undetected until complications arise. And we know that this leads to 2 million deaths globally that are bleeding related, and it has a profound impact on a patient's treatment cost, up to $140,000 incrementally. And for the growing number of DOAC patients that exist right now, there's a significant number that are at risk and are actually experiencing bleeding events, and many that are exposed to invasive surgery also at high risk of bleeding. And so with a tool that can rapidly diagnose bleeding risk, clinicians, or I should say without a tool to rapidly diagnose bleeding risk, clinicians really are hoping for the best, and they hope they see the warning signs before it's too late. And to that end, we have spoken to date to more than 50 clinicians to understand their point of view, and they have validated the market need for new approaches to assessing bleeding risk. And first, they really want to remove the guesswork that they are now encountering as they assess patients relative to bleeding. And they really want information that can inform their decision-making and improve patient outcomes. And also emerging in our discussions are the fact that they are concerned about managing patients that are on these DOAC medications or suspected to be on these medications. And they're particularly concerned about trauma, surgery, and the elderly. And so what they're telling us is they feel as if they're operating in the dark and they have no way to evaluate patients. So they do two things. One, they err on treating preemptively, often giving costly treatments, which may be unnecessary and lead to unintended consequences. And secondly, they don't have the information to actually treat patients and often miss the opportunity, which could lead to additional complications. And so when we presented FlowBio to this group, they see great demand for a test that can understand if DOACs are present in these patients. But they saw even more demand if we could identify the type of DOAC and if we could assess DOAC levels in these patients. And this really sets the stage for our platform technology as well as our first product in development. And I also wanted to share a little background. FlowBio was spun out of the Diamond Lab at the University of Pennsylvania in 2017, and it was really founded on more than 10 years of scientific research by our founder, Dr. Scott Diamond, with over 30 publications and significant pre-company NIH funding. And most recently, they have published a proof of concept data for our first test, the DOAC test, published by Dr. Diamond and our co-inventor, Jason Rossi. And so at FlowBio, we are building upon this work, optimizing the test assay with the goal of generating pilot clinical data in the next 3 months. To that end, we're really well positioned to address the market need, as there's really no good or FDA-approved test that can detect DOACs, the type of DOAC, as well as if a patient is on safe levels and or if they need reversal therapies. And there is none that is accessible and that can provide rapid time to results. It is important to note that there are lab developed specialty tests, although they're labor intensive, they're very complex and they're costly. And because of this, they're not widely available and very impractical for emergency care use. And although there are some coagulation platforms that are attempting to develop a DOAC test, they are not designed to detect DOAC levels, thus are unable to really precisely inform the need for costly reversal therapy. In addition to physician need, hospitals also are incentivized to adopt DOAC testing. First, blood thinners are the #1 drug class implicated in drug adverse events leading to emergency department visits. And this takes a significant toll on the healthcare system, leading to mandates and financial incentives that exist for the focus on safe use, especially as it comes to DOAC medications. And this includes the Joint Commission, which is a hospital accreditation body that has called for testing and evidence-based approaches for the reversal of these DOAC medications. And the FDA has also weighed in, reinforcing the clinical need for such a tool. Now, importantly, hospitals do focus on quality improvement initiatives, quality metrics, and they really make decisions based on reimbursement and the ability to avoid financial penalties and obviously to improve overall profitability. And to that end, in the case of DOACs, we believe that there's a growing incentive that is put into place so that they will avoid reportable events like bleeding and thrombotic events, as well as other downstream complications, and certainly a goal of reducing time to effective treatment. And so in our early assessment, we believe that we are demonstrating alignment of both the clinical need as well as hospital incentives. Additionally, FlowBio has a robust pipeline and we're able to generate multiple revenue streams. Our DOAC test is really just the beginning and our beachhead in the hospital arena, where we're initially targeting in the US over 2 million patients at a $4 billion market opportunity. And we will be generating revenue via a razor blade model. And we also present hospitals, we believe, with a very significant opportunity to take a very tiny step in the first minutes of treatment that can dramatically change patient outcomes while also providing a compelling ROI for the institution. And why wouldn't a hospital spend $100 on a test if they could avoid a DOAC bleed that could cost up to $90,000 per patient, as well as prevent unnecessary treatment, which could cost up to $50,000 per patient. So in terms of development, we've made significant progress and gained traction over the last year. We've raised $964,000 Over 800,000 of that is non-dilutive. We do have a patent pending, and we have earlier this year completed our next-gen prototype build. And we've also been working with former FDA experts to validate our regulatory path and the potential for breakthrough medical device designation. And this really allows us to focus the next 3 months on completing our feasibility work, as well as planning for an FDA pre-sub meeting early next year. And we're currently raising $500,000 and anticipate an additional $13 million needed to get us to FDA clearance. And we're also seeking strategic collaborations to really advance our development program, including our clinical collaborations. And the good news is for FlowBio is that we have secured non-dilutive funding to support our operational runway through the end of 2021. To that end, we are, though, strategically raising $500,000 currently to achieve our value milestones and de-risk our development program to really set us up for success for a Series A raise early next year. And the funds will be used to support our clinical data generation, our cartridge planning, as well as our FDA pre-sub meeting planning. And we are right now considering a convertible note strategy, but are open to other approaches should a lead investor take a key position within the company. So at FlowBio, we are developing an innovative platform technology to remove the blind spot that exists for diagnosing bleeding risk and uncovering the underlying clotting defects. We're developing a platform of tests, and we're presenting a multi-billion dollar opportunity. And we're looking for investors as well as strategic partners to join us on this journey. So thank you very much, and I'd be happy to answer any questions.
Thank you so much. That was a great pitch, Jerianne. First question, I'll actually take the first one. The attendee noted that you're an LLC. Most of our companies start as LLCs. In fact, I guess all of them do. And we convert to a C Corp at the first institutional investment round, or when any investor asks us to. So we'll convert when we need to. Next question, what % of the market do you think that you can capture?
Yeah, so right now we're looking at about 20% of the market, and we will be continuing to do our due diligence with our clinicians and hospitals as we move through the next 6 months to really refine that reflection, but we're currently looking at 20%.
So the next question has to do with the valuation that you expect out of your $9 million Series A.
Yeah, so we have not assigned a valuation for that Series A. Right now we're looking at a convertible note strategy, but certainly as we talk to investors, we are open to have that discussion and move down that path if we can bridge to our Series A.
Okay, any other questions? Yes, we have one more. Can you comment on the applicability to on the battlefield setting, power source, ruggedization, portability, special reagents, etc.?
Yeah, so our goal is to make this as portable and as miniaturized as is reasonable. Our first step obviously is a tabletop, but we do recognize the need, and we're looking to do everything we can as we go through product development through our Series A raise. To explore the boundaries, because it's always a balance between the sensitivity and specificity and how small can you go, but that is absolutely on our radar screen, as well as pre-hospital ambulance marketplace that has also been raised to our attention. So we'll learn more as we go through the next 6 to 9 months.
And if you could talk about your exit strategy.
Yeah, so right now there is a precedent for exit in the diagnostic space, especially for coagulation platforms. So although we are building to obviously create as much balance or value, I should say, and success to commercialize, we recognize, especially with one of the last examples of a company being acquired after their Series A and prior to going into clinical trials, we do realize that's a real possibility. And so obviously we're doing everything we can, as I said, to create the value inflection points and to have options as we move forward.
Great. We appreciate that. So fantastic answers to the questions.
Now presenting will be Eric Corkill, the CEO of Neuralink Technologies.
Thank you, Bhavana. Can everybody hear me? Well, good afternoon, everybody. My name is Eric Corkill. I'm CEO of Neuralink, and I'm very happy to introduce you to Neuralink Technologies, the first automated stroke detection system that is designed to improve hospital outcomes. Strokes are a massive problem in this country, and you're probably already aware of the fact that they are the #1 cause of severe long-term disability, but you may not be aware of the fact that 10% of all of these strokes occur to patients that are already in the hospital. And in fact, less than 50% of these strokes are detected within 4.5 hours of when they occur. And in fact, more than 1 out of 4 patients that have a stroke in the hospital die. So that's hard to believe, but if you understand the contributing factors, it makes sense. First of all, there are procedures that are being performed in the hospital that dramatically increase not only the risk, but also the severity of strokes. Number 2, patients in the hospital, they're bedbound and many times sedated, and this obscures the visual signs of a stroke. And number 3, the standard of care for post-surgical stroke assessment is for having a nurse making rounds. Well, understaffed, overworked clinicians are not able to perform those checks in a consistent and a timely manner. So what does this mean? It means that many strokes that occur are detected too late for the hospital to bring to bear effective stroke mitigation treatments. This increases their length of stay, it increases their rates of morbidity, mortality, and if litigated, has an average settlement cost of almost $1.8 million. So what are we doing about this problem? Well, NeuroAlert is a non-invasive system involving wearable devices placed on patients' wrists, which automatically detect for stroke symptoms. And when they detect those symptoms, the system will immediately alert hospital staff to intervene and speed treatment. This saves lives, this improves outcomes, it reduces risk, it reduces cost. So I'm going to walk you through how the system works in more detail, and it basically has 3 phases. Phase 1 is detection. We use accelerometers which are placed on the patient's wrist, and they're measuring asymmetry in the arm movement of the patient. Asymmetry, classic telltale sign of a stroke. So these smart bands are monitoring asymmetry, and they're transmitting their telemetry to a proprietary algorithm housed in the cloud, which is analyzing the telemetry data, and it's factoring out confounding issues such as, are you right-handed, are you left-handed? Do you happen to have an IV board which is restricting part of your movement? These are the things that make interpreting asymmetry data very difficult, but we have the ability to model those things out and achieve a 95% specificity. Effectively, that's basically 1 false alarm per patient per shift, which is well within the realm of clinical acceptance. So the algorithm analyzes the data, that's step 2, and then when it comes across symptomatic information regarding a potential stroke, it then alerts the clinical staff. In fact, in our first clinical study at the University of Pennsylvania last year on about 200 patients, NeuroAlert demonstrated the ability to detect greater than 80% of strokes in less than an hour. And you have to compare that to what is the current standard of care of less than half of these strokes are detected in 4.5 hours. And because of that time compression, because of the ability to deliver effective stroke mitigation treatments in the hospital, that is our initial target market. We're going to be targeting the medical centers that specialize in high stroke risk cardiovascular procedures and also have neurological teams that are able to step in and provide treatment options to save patients' lives. These are the institutions that have the greatest understanding and the greatest sympathy regarding the problem of in-hospital strokes. Now, beyond that, there are numerous expansion opportunities. Within the acute care marketplace, there are millions of patients that are admitted every single year with high stroke risk symptoms, people with a history of stroke, AFib, congestive heart failure, diabetes, the elderly. So we can expand within the acute care market to that. And then beyond that, non-acute care, international, and even a direct-to-consumer model that ties into existing wearable health monitor devices like Apple Watch or Fitbit, and taps into the digital home assistants. Now, within the hospital marketplace, the pricing for NeuroAlert is a hybrid SaaS pricing, charging a $5,000 per license, per seat license, and that allows an unlimited number of patients to use that seat per year. We'll also be charging $200 for a pair of these smart band bracelets, and they're designed to be disposable, eliminating the cost and the time effort of hospital staff to disinfect or to sterilize. This creates an average per patient cost of only $325. Now you need to compare that to what the hospital is absorbing as cost, because if you stroke in the hospital, your length of stay is extended by anywhere from 2 to 28 days with an average cost per day of $2,000. So looking at the initial target market, these hospitals that are performing 1.7 million high stroke risk cardiovascular procedures, that generates a revenue potential of almost $500 million, and expanding to the other high stroke risk patients that are admitted increases that TAM to almost $5 billion just in the acute care marketplace. Now, there are some competitors out there. We know of at least one that is using asymmetry data as a predictor of strokes. They're also in the incubator stage. However, they don't have our proprietary algorithm that is able to separate the wheat from the chaff. They're relying solely on machine learning and artificial intelligence. And they're not focused on the acute care marketplace. They're focused on SNFs and rehab hospitals. There are, however, multiple billion-dollar, well-established companies in the business of stroke diagnosis and stroke treatment. And we believe these companies will be accelerative to our commercial success, because if NeuroAlert is able to detect strokes faster, then their medications, their technologies, their devices can be brought to bear to save lives and to improve outcomes. So to get us from our current situation, which is starting our second clinical study at Penn, to commercial success, we're looking for $2.5 million in funds to be paid out in 2 tranches. Tranche 1, $500,000, effectively achieves 3 things. We need to finish the design and coding of the backend software. We need to prototype the smart band, which is currently in design by NextFab. And we need to gather the clinical evidence necessary to go for FDA 510K review. And we've already selected a consulting firm to help us in the strategy and the submission process. But the preponderance of the money, $2 million, is really to commercialize the product or the company, to manufacture enough bands, to hire the staff necessary to target the top 100 hospitals by volume that specialize in these high cardiovascular risk procedures. Now, the NeuroAlert team is extremely experienced. The 2 founders, Dr. Messe, Dr. Weimer from Penn. Dr. Messe is a board-certified neurologist and is deeply experienced in the treatment and diagnosis of strokes. Dr. Weimer is a PhD in biomedical engineering and computer science, and is the brainchild trust behind the proprietary algorithm. Our CTO, Det Anson, has years of experience in the Internet of Things, as well as the design and building of wearable devices. And I'm a veteran of the healthcare technology industry, last 23 years, doing startups. NeuroAlert is my 7th startup company, and I've been able to be part of 5 successful exits in the past, totaling almost $500 million. So, folks, we have a big problem, and NeuroAlert is available to address it. We have very innovative technology that is being applied for a patent, sniffing an opportunity, a multi-billion-dollar market potential, positive landscape with limited competition, but with multiple potential partners or acquirers. And we have an experienced team in the medical, clinical, and the startup business acumen necessary to make sure that NeuroAlert is going to be a success. So I thank you for your attention. I look forward to your questions.
Great. Thank you, Eric. And now we will jump to questions. Sorry about that. Computer problem there. Okay. So how protectable is your product?
I'm sorry, how what?
How protectable is the product?
So it's being licensed through the University of Penn. The algorithm has been patented already in a different use case. We're applying for a patent in the use case of stroke detection.
And would you believe that patients might want to use this outside the hospital as well as just inside?
Yeah, I think the growth path of the company starts in the acute care marketplace. It's a difficult environment, but it's also the most concentrated environment for knowing when strokes are going to occur. So we can gather the clinical evidence, build out our business case, generate the revenue, and then begin to look at downstream potentials. But yes, I mean, I'm wearing a Fitbit right now. Effectively, you could wear an Apple Watch Fitbit, put a NeuroAlert band on the other hand. They would talk to one another, and they could talk to your Alexa device and effectively alert EMS services if it detects a stroke.
So you see the NeuroAlert bands as being much cheaper than a Fitbit or an Apple Watch?
Oh, yeah, absolutely. Effectively, it uses an accelerometer, Wi-Fi, Bluetooth, LED light, and a battery, and that's about it. So we're expecting these bands to be able to be manufactured for well under $100 per, which is why the $200 per pair price is being considered.
And what do you ultimately see as your exit strategy?
It depends on the timing. If we stay within the acute care marketplace, there are major monitoring companies like Medtronic and GE that I think would be ideal acquirers. But as you mentioned before, if we expand into the consumer marketplace, Google and Apple are the most appropriate.
And there was one other question of why not use 2 Fitbits? But I think you answered that in terms of that your product will be cheaper than that.
Oh, absolutely.
Okay, well, that's great. Anybody else has any other questions? We might have time for one more. Yep. Have you done a validation study using the same inputs as the first clinical study?
We're just starting the second clinical study at Penn right now. So ask me that question in about 3 months.
Excellent. That sounds great. Well, thank you so much, Eric. That was a fantastic pitch. And he led off. I apologize for everybody. I was out on the first pitch because my computer shut off. But Eric started our digital health, and we'll be moving now to our second digital health company, which will be Joe Camarata with QRS. Thanks so much, Eric.
Thank you.
Hand it off to Joe.
Okay, thank you, everyone. Just a second to share my screen. It doesn't say that I can share my screen. Is there a way for me to share the PowerPoint presentation? Okay,
There should be.
Good, thank you. No, I think I'm good now. Thank you. Hello, everyone. My name is Joe Camarata. I'm President and CEO of Quantitative Radiology Solutions. We're doing body-wide analysis of medical images to inform cancer treatment decisions, and we're commercializing a technology to do automated analysis of medical images from the Department of Radiology at University of Pennsylvania. I'd like to take the time today to talk to you about our first application we have in radiation treatment planning. Radiation treatment planning is used in approximately 2/3 of all newly diagnosed cancer patients. That's over 1 million patients a year that are receiving radiation therapy as part of their treatment. Unfortunately, 1 in 3 of those patients will have a side effect that's serious enough to warrant either an inpatient admission or a visit to the emergency room. And studies have shown that 50% of those side effects are due to suboptimal treatment planning. So we're looking at that treatment planning process and understanding how to remove the causes of errors. One of the areas that we're focused on is something which is called organ at risk contouring. Essentially, when a radiation oncologist creates a plan, they basically have to define the target, the tumor where the radiation should be delivered, but they also need to define those anatomical structures, those organs at risk that need to be protected from radiation. This process is a manual process that's inaccurate, it's inconsistent, and it's time consuming. And while there are some automatic solutions available, our discussions with customers have said that they're not widely adopted because they require too much manual editing. So what we're building here is a solution to automate the contouring of organs at risk. And the technology we have to do that is something that's called automatic anatomy recognition. The best way to think about AAR is its facial recognition for medical images. You give us a CT study, we recognize what part of the body the CT study is in, we recognize the anatomical structures, and then we outline them. So you can see on this slide here, we recognize that it's a head and neck study that was given to us. We identify the relevant structures that needed to be protected from radiation in that study, and then we do the outlining automatically. So the advantages of AAR compared to the existing technologies for automatic contouring is that it generates consistent results based on the published guidelines. Others will use either institutional guidelines or individual preferences, but we actually base our results on medical guidelines in order to improve accuracy. We're able to deliver good results even when we have problematic OARs or studies. We were able to evaluate over 400 cases from the Department of Radiation Oncology at University of Pennsylvania. And what we determined was that 80% of those cases had some type of image quality issue that caused a failure in the automatic contouring solutions. It's no wonder these existing solutions are not widely adopted. And when you bring those two things together, what you'll find is that we'll have less manual editing than the existing automatic solutions. So let me tell you a little bit about what we've seen from our technology so far. We've evaluated the technology at 4 clinical sites. You see them here on the screen. On our behalf, those sites enrolled 214 patients in the study. And we asked them to evaluate the results of our automatic contouring in terms of their accuracy, in terms of their acceptability, and in terms of their efficiency. So let me show you the results from that study. So with regards to accuracy, what the sites have told us is that our contours are more accurate and consistent than clinically drawn contours. We take the one case that was drawn by a human, we take another that was done by our algorithm, we measure the accuracy, and we are more accurate and consistent with the medical guidelines. From an acceptability, which is more of a subjective measure, we simply ask the physicians how acceptable are these results for use in radiation treatment planning. Our contours were as acceptable as clinically drawn contours in 8 of the 10 structures that we measured. And actually the other 2 structures, we just had a misinterpretation of the medical guidelines, so we need to retrain the algorithm for that. But most importantly is the efficiency piece, right? We were able to show that deployment of our technology into the clinical workflow saves an average of 15 to 20 minutes per plan. So, the more accuracy is going to and consistency is going to help to reduce the side effects, but it's the efficiency piece that's going to be really important to drive the adoption of the technology. Just to give you an idea of what those efficiency gains mean, for a clinical site, we kind of thought of three prototypical sites: a non-hospital setting, an integrated delivery network, and an academic medical center. And you can see here the number of patients that they treat. The ability to get an improvement per dosimetrist of 15 to 20 minutes per plan results in quite a number of jump in terms of the new plans and of the new treatments. And you can see if we take just the Medicare reimbursement rates for planning and for treatment, that it results in some significant bottom line improvements for those sites. So the improved accuracy and consistency is targeted towards improving patient outcomes. The improved efficiency is targeted on making it a viable solution to the healthcare provider. So our benefits, in general, we showed that the improved productivity of treatment planning reduces personnel time, that you can use the reduced personnel time to increase revenue by enable addition treatment plans and starting new patients on treatment. And then, of course, what we haven't been able to show yet in our clinical evaluation, but the direction we're heading is to demonstrate that this improved consistency and accuracy improves patient outcomes by avoiding side effects that are common from radiation treatment. So let's talk about the market that we're looking at. For this first application of our technology, this radiation treatment planning that I mentioned, it's a $200 million annual market. As I mentioned, there's more than 1 million patients a year that are treated with that. But we actually play in a larger market. We play in the $1 billion market for automated analysis of imaging in cancer care. And we recently were awarded a second grant from the National Cancer Institute that allows us to expand the technology from the first application, which I've been describing to you, to a second application in the area of medical oncology to begin to monitor treatment response. As we look to get into market, what we're going to focus on are the proton centers. Proton therapy is an advanced form of radiation therapy that delivers very, very high doses of radiation to precise points. These centers have the most acute need for a highly accurate treatment, but also an efficient one that allows them to replan frequently during the course of treatment. There are, as you can see, 36 of these proton centers in the United States. We already have relationships with 4 of them, and through our advisors have begun to build out relationships with the others. These will be the early adopters of our technology. We're currently raising a round of $1.8 million to launch our product. We submitted the product yesterday for FDA clearance. We expect to have that clearance in March of 2021 and be able to start generating revenue at that time. The round that we're raising now is to support that product launch. $1.4 million of that round will be used for commercializations and operations, getting the product into market and support it when it's available to the customers. We've also reserved some money to do product enhancements to further integrate the product into the clinical workflow. We built this product and submitted it to the FDA largely through non-dilutive funding. We've had $2.5 million of grants coming from NSF and NCI, National Cancer Institute, but we've also raised an additional $500,000 in convertible notes through the Phase 1 Ventures at the Science Center and also Ben Franklin Technology Partners. So this is our overall go-to-market roadmap. As I mentioned, we expect our 510(k) clearance in March of next year. We expect to then do release for sale and start generating revenue at that time. And the money that we're raising will be used in order primarily for business development and then for product enhancements. So with that, the management team, forgot. So myself, I have a background 30 years plus in medical imaging at Siemens and GE Healthcare, largely in the commercialization of innovations of medical imaging. Our Chief Technology Officer is Stephen Owens, and Steve and I work together at Siemens. Steve actually started the U.S. software development organization for Siemens Healthcare in the United States in Princeton, New Jersey. He has tons of experience of bringing to market products in a regulated environment. And our 2 co-founders are from the Department of Radiology at University of Pennsylvania. Dr. Adupa is a computer scientist, Dr. Theresian, a radiologist, still extremely involved with the company and working with us on the grants that I mentioned before. So with that, I think we have a great opportunity to begin to impact patient care and radiation therapy, especially to reduce side effects. We have a strong economic value proposition to drive adoption of our products. We are waiting for our FDA clearance and to start generating revenue in 2021.
Okay. This is great. Thank you so much, Joe. Fantastic presentation. What cancers are you going to focus on first?
So very good. The current cancers are head and neck cancers and thoracic cancer, so lung and breast cancer.
How does your product compare to ViewRay mRidian technology?
So ViewRay actually, so we are using CT images for doing our treatment planning. ViewRay is an MRI machine. It's a combined MRI and radiation oncology. So they're a hardware product. We're just a software play. We have shown the technology works also with MRI, so we could in the future apply it to ViewRay planning.
Have you thought about tumor contouring versus organ contouring?
Yes, actually we have. In fact, I mentioned there is that second grant that we have from NCI, and that's actually allowing us to recognize and begin to quantify diseased tissue as opposed to healthy tissue that we're looking at here. And that will be kind of the precursor to us to be able to do tumor contouring.
Which academic medical centers have you been working with?
Well, University of Pennsylvania, Washington University in St. Louis, Rutgers University, and the New York Proton Center up in Upper East Side, which is a consortium of Memorial Sloan Kettering and Montefiore Medical.
That's excellent. Does anyone else have any other questions? All right, if there are no other questions, thank you so much, Joe.
All right, thanks.
I really appreciate the presentation. It was excellent. And we will be moving next to our company Cogware, and David Yance will be the presenter. And here comes David.
Hey, good afternoon.
Oh, and David, because we're running this on a strict timeline for everyone, we're actually managing that we're a couple minutes ahead.
Okay.
So I just want to be careful that we don't have somebody join on at 2:15 to hear your pitch, and then you'd be 3 minutes into it. So maybe we'll just pause for just a second to make sure that we give everyone a chance to get here on time.
Okay, very good.
I applaud all of our previous speakers that everyone is finishing on time, so we're able to keep right to the letter, which is great.
Michael, can you see my slides fine?
I can.
Excellent.
And I applaud your background as well.
All right, thank you.
Very professional. I'm gonna give it one more minute and then we'll kick off. It'll give you an extra minute. I can point out as we're getting ready to start the next pitch that we will be sharing the recording next week. So if anyone misses any part of a presentation or a whole presentation, they will certainly be able to catch up on it next week. As well as anyone else who is unable to make it today, we'll be able to get the presentation to them. So with that, let's kick off with David Yance and Cogware. Take it away.
All right, thank you very much, and welcome everybody. I appreciate you making time today. I'm excited to talk to you about Cogware and our first application for a next generation cognitive wearable. Hold on. There we go. You're likely familiar with the wearable trend that's occurred over the last decade, and it started with essentially devices that could measure motion and heart rate. Since then, it's been expanded to also measure heart rate variability. And newer devices like the Apple Watch or the Whoop band are using heart rate variability to also measure things like sleep quality, stress, and athletic recovery time. Now, the issue with this is that heart rate variability is a very non-specific measure. So what we're going to do is measure brain waves in order to get those measures directly. Now, beyond that, we can also gather other cognitive information about how the brain is functioning, things like excitement, engagement, focus, or distraction. And this really gives us now the ability to think about solving unmet needs in different ways and opens up the ability for us to impact across the diverse areas of industries, from healthcare to industrial safety to business marketing. But today, I'd like to talk to you about mental health. You can hardly get through a day in the news today without hearing about the mental health crisis we're having from COVID-19. However, even prior to COVID-19, mental health is a huge issue. If you think about it, just worldwide, only focus on anxiety, there's already 264 million people who suffer from anxiety. Within the US, 1 in 5 adults suffer from a mental disorder, and 15 million people annually are seeking treatment for anxiety in the US. If you're one of these patients, you're likely going to encounter a therapist who already has a very large patient burden and a very heavy workload. They're also likely going to be very tired. One of the trends that we've noted during our market research is that our therapists are telling us that they're very fatigued from telemedicine. The reason for this is because during the therapy process, it's a very qualitative process, and they're picking up nonverbal cues. Now with telemedicine, that's much more difficult, and they only see a snapshot of the patient. All of this comes together really to bring up an issue that there's very limited patient trending and quality metrics in general for mental health. And this is an issue that's impacting more broadly. Now, imagine, if you would, the ability, much like a flu patient would be able to take their temperature to trend how they're doing from the flu and see when they're recovering, or a heart failure patient taking their blood pressure on a daily basis to try to predict decompensation, that you could have the same type of biomarker for mental health. And that's precisely what we are going to be bringing to market. We're going to be bringing to market the first biomarker for anxiety, and we call it our anxiety thermometer, that's able to measure and trend anxiety over time based upon the underlying brain biology. We have 2 pieces of intellectual property that allow us to do this where others have struggled. The first are our nanotechnology sensor electrodes. Compared with other electrodes that may provide very high clinical grade signals for a limited amount of time or provide convenience, but really don't provide the clinical signal quality, ours present the best of both worlds. Our sensors have mechanical properties that very closely mimic skin, very high conductance. And essentially what this means is that we can get clinical grade signals for as long as you want in just about any application that you want. The other part that comes along with this are neuro-targeted algorithms. And these are algorithms that have been in development for close to a decade. And they specifically target the anterior cingulate cortex, which is a part of the brain that regulates anxiety and depression. To date, these algorithms have been validated with close to 1,000 people, and they've been shown to be over 85% accurate. We've also, there's also been studies in cross-cultural environments in that accuracy is held. And currently there's an arm of this research that's undergoing with severe depression patients undergoing transcranial magnetic stimulation. I'd like to talk a little bit about the architecture and how this might work. Our architecture has 3 main components: an EEG headband, a user mobile app, and our HIPAA compliant data platform. So essentially the way this would work is a user would wear an EEG headband at particular points during the day and interface it with our mobile app on a mobile device. This mobile app will collect EEG data. At the same time, it will prompt the user to play a particular game that's part of the algorithm. This data is then sent up to the data platform where the data is crunched and an anxiety score is created. Now, we're developing this to be HIPAA compliant. That will also give us the ability to de-identify the data and essentially perform other big data analysis on it in the future to develop new algorithms. Plus, this has the potential to grow into one of the world's biggest brain databases. Much like the user mobile app, the provider and the clinician also has a mobile app and a portal that allows them to trend the patients over time, and potentially, if a patient is trending in the wrong direction, prioritize them for being seen and essentially get them back on track of therapy. We've done 2 rounds of market research with clinicians, both prior and after COVID-19. And we've gotten very, very positive feedback to this concept. Right away, the clinicians saw the unmet needs that were met from just having a physiological anxiety marker, it allowing them to help triage and trend their patients, especially with the large burden of patients that they have. And also they saw that this could be a welcome addition to telemedicine to give them more insight into their patients. What's even more exciting to us is that when we started talking to them and roll this out, they started coming up with even more applications than we had initially thought of. So for instance, many saw this as a way to quantify the response from pharmaceuticals. Today, that's very much a trial and error process, and there's really no measure to quantify how well pharmaceuticals are working. And then also another very important area is with children. Therapists who deal with children have an added burden that children can't express their feelings nearly as well as adults. And in fact, because there's no real clear quality metric, that's one of the reasons that children fall out of therapy prematurely. So that's an area where they're excited about employing something like this. Now, anytime that you're bringing a healthcare product to market, certainly the clinical outcomes are key. You have to deliver that. But in today's market, healthcare economic benefits are also critical for adoption. Because we are bringing a physiologic measurement to market, our system is going to fit within existing Medicare codes that have been used for other neurological brain conditions for remote monitoring. The way we're going to price this is to fit it within an 8 to 10 week anxiety therapy session. That's the basic session that people start with. And because of this, the clinic is going to be able to have revenue come in at a decent margin. We're going to be able to price our system such that we bring revenue in at a decent margin. So essentially, we're providing benefits to the patient, to Codware, and to the clinic. As we think about our product development process and the way that we're architecting the system, we really have 3 value inflection points in order for us to get to market. So to date, we've developed pilot headgear with our novel nanotechnology sensors. We have anxiety algorithms, as I mentioned before, that have been validated in nearly 1,000 patients. And we've shown proof of principle of data transfer from our headgear to the mobile device up into the cloud. The first $100,000 of investment will be used to miniaturize your electronics so it can fit into a smaller form factor. $500,000 of investment will enable us to develop our next generation headgear. While what we have today works, what we're going to go for is a smaller and more compliant form factor, one that's easier to use for the average user. And we're also going to develop this under FDA design controls so we can obtain Class 2 clearance from the FDA. $2 million of investment will bring us both of those hardware components and also allow us to develop our HIPAA compliant platform. And as I said before, this is where all the data is going to be hosted. And we're also going to develop our mobile application at that point too. Now, when we get to this point in our development process, we know we're going to have to raise another round to expand manufacturing, to scale up manufacturing, and also to build out a commercial organization. We do have in line a potential clinical study for adoption. And I want to be very clear on this, that this is based upon customer feedback, that this is only for adoption and not for FDA clearance. But what's very important at this point to realize is once we are here, we essentially have a platform. And launching into new applications is only a matter of changing the algorithms that are run on the platform in the front end of the mobile app. So we can very quickly, rapidly prototype for new indications. Perhaps the lowest hanging fruit of these are just the research grade device. We get regular inquiries about whether our device could be used for a whole host of academic and medical research. And currently today, this is a $472 million market. So after we spent the $2 million and we have our product developed, we can quickly target the research market as well as the anxiety market. The financials of this are very attractive. So all in, including the development costs, a potential clinical trial, the launch expenses, marketing and sales growth, and commercial organization, we see ourselves breaking even from a cash flow basis in less than 5 years. And this occurs with us penetrating 3% of the research market and far less than 1% of the overall anxiety market. The way that we like to think about this is essentially $2 million in product development is going to create and unlock $6.5 billion of market potential given the revenues I showed you a couple slides back. And of course, we have upside in many other applications as well. Before I finish, I'd like to talk about our team members. We're basically a group of experienced neuroscientists and executives. Dr. Michael Platt is one of the co-founders. He's a neuroscience pioneer and with a triple appointment at Penn. He is the director and founder of the Wharton Neuroscience Initiative and the inventor of our neurotargeting algorithms. Arjun Ramakrishnan is the other co-founder. He's had appointments both at Penn and Duke and currently is on the faculty of the Indian Institute of Technology. And he's the inventor of the nanotechnology electrodes and also one of the key researchers of our anxiety algorithms. Myself, I'm a 20+ year veteran of the medical device industry. I've repositioned companies for growth and prepared them for acquisition. And I've also along the way helped deliver over 50 products to market. And you all know Michael Poisel, so we're thrilled to have him on our board and the advice that he can provide us as well. So in summary, I hope you'll consider joining us. Our anxiety thermometer, our first application, meets multiple unmet needs, fits in with existing reimbursement, and can help up to 264 million people today. $2 million in product development is going to open up $6.5 billion in healthcare market opportunity in the US alone, and we expect a cash flow break even within 5 years. We're architecting this platform for rapid customization, quick initial path to revenue, and future optionality. And finally, as I talk about those other avenues we can get into, we're already thinking about and documenting IP that can be used for many different other big societal needs. So with that, I'd like to thank you for your time and certainly happy to entertain any questions.
Excellent job. Thank you, David. We will now check our questions. So the first question is, can you talk some more about the accuracy and consistency of the data that you get from the product?
Yeah, so far we are still in the integration of the product. Right now, the specificity of the outcome of the algorithm is over 85%. I think doing a deeper dive, I'd be happy to take that offline, and I would love to bring in our co-founders for that discussion.
That's great. And what do you see as your exit strategy?
There are a couple different exit strategies that we are considering. We've already been in some initial discussions with mental health companies that have digital platforms. Obviously, this could be a very nice addition to their platform. We could potentially build it out by ourselves as well, but that's something that as the mental health market continues to evolve, we're going to evaluate.
And what about in terms of different markets that you plan to pursue? Are there markets planned beyond mental health?
So within healthcare, mental health is the first one. Some of the other ones that come to mind that are fairly unique, certainly within the military, as we think about team dynamics and we think about the same type of healthcare avenues. So there's certainly anxiety within the military, but there's also treatment of PTSD. So fairly unique. And so we're looking at those avenues there. And then ultimately, another area that's interesting to me is just industrial safety, because this gives a different perspective in terms of operators in highly critical operations.
Are you open to angels investing or just a institutional round?
We are open to angels investing at this point.
Great. So unless anyone else has any further questions, have you thought about the terms of the round that you're willing to offer?
That's something that we're still working out. At this point, we are early. We haven't placed evaluation on the company, so a lot of the terms are ultimately going to depend upon where we end up there.
Excellent. That's great. So thank you so much, David. Fantastic pitch.
All right. Thank you.
We will be moving next to our therapeutics section, and we will be starting that with Pinpoint Therapeutics. And Christian Peters will be up shortly. I appreciate everybody sticking around for another section of our presentations today. And with that, it is 2:28, Christian, so we'll have you get ready your pitch so that you'll be starting right at 2:30.
Okay, very good. Okay, so do you see my screen, Michael?
I do.
Okay, great. Hello, everyone. Thank you for letting me participate in this opportunity. Pinpoint Therapeutics is a relatively new company, and I'd like you to walk through what we've done so far and also what we're asking for in next round of financing. Here, just an overview. Pinpoint is developing novel autophagy inhibitors for the treatment of treatment-resistant cancers in mainly melanoma, pancreas, and colon at the moment. We're not limited to that, but we have the best data in these areas. We were founded in 2018 with the technology licensed from the University of Pennsylvania. And at the moment, we have one full-time employee, that's me, with 2 faculty co-founders who are also heavily involved. Our technology is a first-in-class autophagy inhibitors to overcome resistance to current therapies for melanoma, pancreas, and colon cancer. And we have shown so far, and I will show you some data about this, that we have single agent activity. It's quite robust, actually. We also can combine our drugs with chemotherapy that is on the market already. And quite exciting, there's an ability to combine our MOA, our drugs, with the emerging immune therapies. And I'll just talk to it a little bit. I don't show you any data, but this is the latest development. And we have data that shows that we are effective in multiple cancer cell lines. We've tested 50 so far, and we have good sensitivity for all these cancer cell lines in various therapeutic areas. We're seeking $3.5 million for the second round. We have an initial round from PCI Ventures for $1 million, which we have used to generate the data that you will see. And I have a preliminary phase 1 estimate, which runs between $11.5 and $13.5 million if we want to go down that road. Here's the team. Ravi Amaravadi and Jeffrey Winkler are the co-founders and the inventors of the technology. They're both faculty, and they're also part of the management team of Pinpoint. I myself, I have an MD, PhD as a background, have worked for 30 years in development, 10 years preclinically, 20 years I've been in companies and have done 7 FDA approvals, phases 1 through 4, and have also done commercial launches in the recent past. Here, a quick overview of what autophagy is. Some of you have heard about autophagy, others have not. In general, it's a resistance pathway that cancer cells or cancers will turn on when they're under stress. So chemotherapy, radiation, all will induce autophagy, and it leads to repurposing of cell debris into nutrients that the cancer can reutilize to grow again. So when you look at the lower panel here, in the system where we have chemotherapy or radiation, cancer cells are dying initially. Once they activate the autophagy pathway, we can inhibit this pathway quite effectively, which then will lead to a dramatic reduction of cancer growth, and I will show you those data. Just a quick overview over the technology. The molecules that you see here on the left side are the 2 molecules that have been published in 2017 and 2019, which led to the discovery that these dimeric hydroxychloroquine and quinacrines, that's what the technology is based on, will lead to an effective inhibition of a new target, an enzyme inside the lysosome of the cell. And that inhibition leads to a dual mechanism, that a growth factor that helps survive cancers, mTOR, as well as the autophagy pathway itself is inhibited. So we're dealing here really with a dual pathway MOA that helps drive the success of the molecules that we've seen so far. When you look at the ability to reduce cancer growth, there's one of the graphics from Ravi's initial paper from 2019. You can see very nicely the reduction of cancer growth here in red as compared to the control group when he has used one of his molecules that are published in a human colorectal cancer cell line. So it's a human xenograft in a mouse system that has led to a dramatic reduction of tumor growth. This is also the finding that led to the founding of the company. And with the initial $1 million of seed round funding that we got from Kairos Ventures, we have now made 12 compounds, all variations on the theme that Ravi and Jeff have published. And we have run a number of DMPK and ADMET studies that you can see here in the middle in the green. So since January of this year, we have been able to generate data with our new molecules that look quite exciting. So for example, an IC50 range in the micromolar ranges with our molecules between 7.3 and 0.04, quite a sensitivity that we didn't expect to see to that degree, with the majority of the molecules being roughly under 2. We have developed a number of data that I won't show you here, just a overview of what we have done so far. We recently have gone into maximum tolerated studies in tumor models. So I will show you one of our tumor models and the result of it, which came out of a marine melanoma B16F10 cell line. So we've used in this experiment multiple doses ranging from 0.5 to 20 mg per kg. And you see on the right here that the regression of the tumor growth, or the reduction of the tumor growth, follows very nicely the dose range that we have given. And we're quite excited. Of course, the medical community is quite excited to see such strong single agent activity. To date, there are only a few autophagy inhibitor in development, and none as of today has shown single agent activity. So we can say that with a high degree of confidence. We just had a conference that was hosted also virtually, like this event, which made it very clear that at the moment, we're the only game in town with single agent activity and a robust result of this nature. In the meantime, we also have the initial A375 cell line melanoma in humans, where we see similar results. IP position is quite strong. We have, just an overview here, 4 patent families ranging from at least LOE 32 to 2040. And we are, at the moment, we have an exclusive license option with Penn. We are in the final throes of getting the full exclusive license from the University of Pennsylvania. A quick look at the market size. It is in cancer and expected to be high, but I just wanted to show that we've done, in cooperation with Wharton, an overview over the indications that we're most interested in. And you see here essentially the implied total market potential with quite a wide range and of possibilities here in this area. And particularly, we see very high sensitivity of our drug in pancreatic cancer. Yes, it's the graveyard of clinical development, but we also see with our particular MOA very, very high efficacy in this area. Here's just a breakdown of the funds to IND. Essentially, we have done a good amount of initial work to see that we have an effective molecule. We would need to have more done work in order to speak to the FDA. And I won't read you the list here. Many of you have been in this business for years, so you understand there is the IND enabling studies. There are the IND enabling studies that need to be done, especially with a new type of formulation. We have pre-formulation, but we don't have full formulation, so we would need to develop the CMC file and the IV formulation in particular. Just to give you a current status and recap, we have done the biological characterization of 12 compounds. Many of these compounds are in the feasible range to be further developed. We've done initial metabolism tumor studies. We've done a preliminary selection of leads, and we have found a good dose range, which allows us to also think about combining with other chemotherapy or immunotherapies. The main things for the next step are really the formulation and CMC development, the GLP talks that we need to do, and of course, start partnering with the FDA on regulatory approaches. That includes the clinical development. And the funding that we're seeking for is indeed CMC, GLP formulation, IND enabling tox, and DMPK admit studies. So in summary, we have a new target in the autophagy pathway called PPT1 that provides an exciting new approach to overcome therapy-resistant cancers. We anticipate that our PPT1 inhibitors will be effective in multiple cancers, not only colon cancer, pancreas, and melanoma, but others as well, but we need to look further into the possibilities of that nature. Our compounds show robust single agent activity. They're not immunosuppressant from what we understand at the moment, and they can be combined with chemotherapy. So we need to plan for IND enabling studies, aiming for 3.5 million to get to the IND. Timing would approximately be 14 months from funds available. There is some room for doing things faster, but as you know, promises are made easy, and then the execution is really something that we need to focus on. So I have, from my consultant and preliminary discussions with CROs, to get to IND a year, but we do need to build in a little bit of a buffer there. So if funding was to do, was not in May 22, but sorry about this. So if funding were to come around in 21, we would be within IND in June 22, if no obstacles are encountered. And we have already started with plans on a phase 1 trial that I can talk to you a little more if there are any questions. And that's it.
Great. Thank you so much, Christian. Now we have a little bit of time for some questions. Can you talk about your IP position and whether the compounds are protected?
Yes, so we have 4 families of IP protection, not only composition of matter, but also treatment. And we have ongoing IP filings. So at the moment, in a best case scenario, we're protected out to 2040.
That's great. Are there any corollary treatments out there that would give you an idea of what the side effects and this might be with this technology and these drugs?
The best corollary would be the use of hydroxychloroquine in addition to cancer treatments, which has been done for the past 10 years very successfully. And there have been no reported outcomes. But I need to caveat that because when you use an approved medication, you don't collect all the AEs. We expect to have a high degree of safety based on our molecules, but also on our recent findings that we've done. We've done some preliminary toxicology, and that looks extremely promising.
Excellent. Are you still open to angels, or have you moved into venture capital only?
No, open to angel.
Okay. And just to clarify, your IP currently is licensed from Penn. It's not owned by Pinpoint yet, right?
That's correct.
Have you identified a lead investor yet?
We do not. We have our current investor, PCI Ventures, and they are interested to stay in contact with us. But they also have signaled that for the next round, they'd like to broaden the field as well.
Excellent. If there,
we have one more question in the chat. Christian, what is the term for the current raise and the valuation?
So we have not done a valuation at the moment. We're in the middle of doing a valuation. We have had no data, and we're completely novel. That is just in the beginning, and we would need to talk about that some more.
Excellent. Thank you so much, Christian. A fantastic pitch. And we are moving next on to Epivario. And we will have Thomas Kim, who's the CEO of Epivario, will be joining us to give that pitch. How are you doing today, Thomas?
Hey, great. Thanks, Mike. Appreciate this opportunity. And I got to say, there's been some pretty outstanding pitches. So definitely honored to be a part of this community. So let me share my screen. Okay. Well, thank you. I'm here to talk about Epivario. At Epivario, we're just establishing a revolutionary treatment to treat the root cause of addiction and PTSD. About Epivario, it's a spin out, as the other companies here, of the University of Pennsylvania. We're expanding on this breakthrough discovery of an epigenetic gene regulation that establishes some memory formation. And through this, we're developing a small molecule inhibitor to treat memory-related psychiatric disorders. And the core discoveries are published in Nature back in May of 2017 and October of 2019. And recently, earlier this year, we were a finalist in the Nature Spinoff Prize. And you can see the link to the publication here. The company was founded by Shelley Berger at Penn, Director of Epigenetics, and Philip Muse, who's currently a postdoc in Dr. Eric Nessler's lab at Mount Sinai. And myself, I am also an entrepreneur, recently with Inovio Pharmaceuticals, another University of Penn spinout. We've been fortunate to have a strong scientific advisory board, including the founders and some renowned neuroscientists and epigeneticists, and been fortunate to establish some academic collaborations with some leading experts in the field of stress models, anxiety, and addiction. And really what we're looking at is attacking and providing new solutions to large markets with an exacerbating and growing need for new therapies. If you look at PTSD, about 8% of the population over their lifetime will experience PTSD. In the US alone, nearly 30 million are afflicted with this disease. And right now, the treatments available are very limited. The standard of care is psychotherapy, and there's only antidepressants really to treat on the pharmacal side. And what we're really doing is we're providing a drug to limit memory reconsolidation. And what we're trying to do is disassociate the stress, the physiological response to the traumatic memory. And the core of the discovery is really this enzyme, the discovery of acetyl-CoA synthetase as a enzyme that regulates through converting acetate to acetyl-CoA and providing that as a source of fuel for a histone acetylase that upregulates early memory genes that are necessary for memory. And the proof of concept that we've established in a behavioral model for PTSD is with rats, you expose them to a stress stimulus, here cat urine, and subsequently you re-expose them to a cue or context that triggers that memory, and then you measure their motion. And the greater the amount of freezing, the greater the stress experienced by the animals. And you can see with our drug cohort, there's a substantial reduction in the amount of freezing in the animals. And how this translates into a human therapeutic is often in the psychotherapy treatments, there is guided recall where, through your therapist, you remodel the experience that you have when you recall the traumatic memory. And we feel at this time, if you can start manipulating and disassociating the stress that's associated with the traumatic memory, that can provide a healthy state for you to then go through your cognitive therapy with your psychotherapist. In addition, I'll move to addiction. As everybody knows, addiction has been pretty prominent in the media, especially with the pandemic of late. Greater consumption of alcohol and other drugs. It's a huge burden to society. You can see the overall spend that we have towards healthcare related to these various substances of abuse. Here's a title from our publication back in 2019 about alcohol metabolism, how that triggers this enhanced memory related to drinking alcohol. And you can see in this cartoon the progression of alcohol metabolism, and then the ACSS2 converting to acetyl-CoA, and then regulating this histone acetylation, this epigenetic regulation of early memory genes. And the initial proof of concept that we established, and which was published in that paper, is through this animal model, condition place preference. Here you can see the animals are conditioned to alcohol on one side and then saline on the other side. And after 8 days, they're given a choice test. And here you can see the data with wild type, the increased blue, you can see a preference towards the alcohol side. And this was done with a genetic knockdown of ACSS2. We show no preference. And this is not published, but in our co-founder's lab, they repeated this experiment with cocaine. And here it's graphed a little bit different, but you can see the relative time in each chamber. So here, wild type on the left, you can see on the blank side, the white bar graph, you can see an enhanced amount of time spent on the cocaine side, whereas with our genetic knockdown, they're about even. So right now we are still working on developing PTSD therapeutic. We're in the process of applying for an SBIR grant with the National Institute of Mental Health. On the addiction side, we've established collaborations, and we're working with Chris Cowan at the Medical University of South Carolina on an addiction model, self-administration addiction model for cocaine, and then Paul Kenny at Mount Sinai for nicotine self-admin model. We've exclusively licensed 2 patent families from the University of Pennsylvania related to the method of use and some novel compounds. We've been fortunate to have a array of very experienced business advisors, including Mike Dishwitz at PCI. To date, we've raised a little bit over $700,000 through this. We've completed an SRA with the University of Pennsylvania with our co-founder. We hired a full-time researcher, and we were able to complete a lot of preclinical milestones, including some of these proof of concept studies and some DMPK work with Charles River Labs. Currently, we're looking to raise $15 million in the middle of 2021. This would definitely be triggered, particularly on the heels of positive data in the self-admin study for cocaine addiction. And this would fund PTSD in one part, addiction on another, and then some core R&D to develop a further behavioral models and develop a biomarker as well. And that's the end of my presentation. I appreciate the opportunity to talk to you about Epivarium.
Excellent. Thank you so much, Thomas. Great pitch. Now we'll check out the questions. see. So how would the drug be delivered?
We're working on an oral formulation right now. The drug has been delivered IP currently in the animal models.
And considering you're trying to raise 15 million, are you interested in angels or have you moved straight to VCs?
We just closed a bridge, bringing in some angels. If it's an angel group that's interested and would consider participating as part of a syndicate for the Series A, we would consider that.
Okay. In the context of PTSD, how long after a traumatic event could this be treated and implemented and still be effective?
Yeah, so for PTSD, we would be looking to treat patients who've relapsed, who've undergone standard of care and relapse and is looking for treatment, further treatment. That's what the patient population would be.
Mm-hmm. Have you thought about the terms for your next round at all?
We have not. We've done sort of back of the napkin, kind of pontificating, I guess. But I honestly, it would depend on the strength of this cocaine self-administration study that we're currently going through right now.
How do you view the treatment in terms of, is it a short-term, a mid-term treatment, or is this something somebody have to take for the rest of their life?
No, this would be right now acute therapy. It would be a augmented psychotherapy session. So it would be a first phase of cue recall with your psychotherapist while on our drug. And then you would follow up with cognitive psychotherapy.
Would you consider allowing an angel to invest in the bridge that you just closed? And if so, what were the terms?
Yeah, again, it's the same I said before. If it would be an angel group that would consider coming in on the follow-on, and the terms would be a 20% discount on conversion and a 4.5 million pre-money cap.
Do you think that even in the case of alcoholism, that it could still be a short-term treatment?
Yes, and the way it would work, and we've been working with our clinical advisors on this. We're working on limiting or eliminating cue-induced addictive behavior. So if you're trying to abstain and you're worried about cue or context bringing up craving memories, that's really what we're trying to eliminate.
That's excellent. Anyone else have any other questions? I think that's it. If that's the case, thank you so much, Thomas. Excellent pitch.
Thanks,
And
Mike.
again,
Appreciate
if
it.
anyone is interested in further conversations, you can reach out to Thomas directly or to anyone else on the team. Great. So next up on our list will be Interface. And
great, thank you.
Nader will be coming up to pitch. Bringing him up now. There's Nader. Great.
Thanks,
excellent.
And
And we're 1 minute ahead. So if you could just take a minute to share your screen. We'll get you set up to start at 3 o'clock.
Great. Can you see my screen?
I can.
Great. Okay.
And off you go. Thanks so much, Nader.
Great. Well, thank you so much for the opportunity to present to the team here. I'd like to tell you about our company, Interface. And as the name implies, Interface's goal is to re-innervate lost neuronal circuitry through anatomically inspired microtissue that really looks to recapitulate aberrant circuitry rather than just the signal that's lost when a particular circuitry happens to degenerate. Our lead asset is in Parkinson's disease. And in Parkinson's disease, we're currently pursuing IND-enabling studies. And The ask from this group would be a $2 to $3 million raise, and that would be to support both definitive efficacy studies and some early safety studies that would allow us to have a regulatory engagement in the latter half of 2021, which would hopefully set us up, given some positive feedback from the regulatory agencies, to go out and raise a strong Series A. We've currently raised funding and some seed funding that allows us to operate through Q2 2021. While Parkinson's is our main indication, we do have other indications that we're pursuing, and we're pursuing these indications as proof of concept in a number of other indications within the central nervous system, though this technology could also be applied in the periphery as well. This technology was developed by Dr. Casey Cullen and Doug Smith at the University of Pennsylvania. So stem cells have been around and have been a therapeutic potential and a therapeutic promise for nearly 40 years. But what we've seen is that promise has been largely unrealized for a lot of particular disorders, and in particularly in the central nervous system. And one of the reasons maybe is redepositing cells which have been lost, so that stem cells alone may not be enough to exert a therapeutic effect, and that what we need is the combination of cells plus scaffolding, or cells plus a therapeutic context or milieu, to really realize the full potential of stem cells in regenerative medicine. And the bioengineered, anatomically inspired tissue pathways that we have created really do look to recreate that lost circuitry. And when I say anatomically inspired allows us to customize the type of cell, the number of cells, and the encasement as well, so that the biofabricated pathway really does have the direct context for the particular indication. So there's a lot of customization that's available that allows us to customize the right type of circuitry for the right type of indication we're looking to pursue and to ameliorate the effects of. These circuitry pathways can be constructed so they're unidirectional or bidirectional signaling. And again, we can customize the length, the dose, and the cell type and the cell source, all of this based on the indication. And once constructed, we believe that these implants can be implanted via minimally invasive MRI-guided stereotaxic implantation. So Parkinson's disease, as I mentioned, is our lead indication. And that really is a hand-in-glove fit for this particular technology. And the reason is that Parkinson's disease is a very well-characterized disease with a very well-known pathology. We see a lost circuitry between the substantia that originates in the substantia nigra and terminates in the striatum. And the termination in the striatum is where we see dopamine release. And once this dopamine is, once the circuitry is degenerated, we no longer see circuitry within the striatum. So having a very well-defined pathology and a very well-defined pathway is really the perfect candidate for Innervace's technology. There are no disease-modifying therapeutics available for Parkinson's disease, so there's a huge unmet medical need from a patient perspective, from a caregiver perspective. Parkinson's disease is the second most common neurodegenerative disorders, and patients can often suffer from Parkinson's disease for decades or more, which results in a huge economic impact to health systems. So from a patient perspective, from a caregiver perspective, and from a health system perspective, again, as well as the pathology, Parkinson's is the ideal application, lead application for Innervace. And the idea of using regenerative medicine or stem cells to restore lost circuitry, particularly in Parkinson's disease, is actually not a new concept. It's almost a 40-year-old concept beginning in the 1980s, where there was grafting of fetal midbrain tissue into the striatum. And again, the striatum is where the signal terminates, not where the circuitry originates. But the early studies showed some interesting efficacy and really served as a proof of concept that there could be a durable long-term clinical benefit from stem cell implantations. But the biggest side effect that was seen was the uncontrolled dopamine release, because again, you're adding the signal where the signal acts rather than recreating the circuitry, resulted in a lot of side effects for patients. And moving from the 1980s to where we are now, we have seen some fits and starts of stem cells being used as a regenerative medicine approach for Parkinson's disease. And we're currently at the point where we are seeing a lot of clinical activity and a re-engagement of stem cells for the treatment of Parkinson's disease. But again, these treatments are still basically putting cells where the signal is lost in the striatum rather than recreating the circuitry. So it really stems back, and there's an opportunity where, while this may produce some disease modifying effects for Parkinson's disease, you still do have a really big chance of having these unintended side effects that we did see in the treatments that were initiated nearly 40 years ago. But again, Intervase's technology looks to recreate the circuitry by not only adding dopamine where dopamine is lost within the striatum, but having that connection between where the signal ends and where the signal begins within the substantia nigra. So it's recreating the circuitry, not just restoring dopamine, which stem cells can do, and also current pharmacological treatments look to do as well. Here you can see some tissue engineered pathways that have been implanted into rodents. And what we see is these implants survive. This is looking at a time point for 1 month. They also do extend the appropriate innervation into the striatum that we can see here. And they do also integrate into the substantia nigra. So we do see midbrain to forebrain connections where these implants, once created in a lab and implanted it into an animal, really do integrate. So the circuitry integrates and then can have a reliable signal based on inputs and outputs from the midbrain and the striatum. So while this data we're showing here is currently at 1 month, Dr. Cullen and Intervase is working on generating additional cell survival data for a 6-week time point and a 3-month time point. Aside from the anatomical restoration of a lost circuitry, we also do the functional implications of restoring that circuitry. So while dopamine is lost within the striatum, what we do see within the animal models of Parkinson's disease, this is some voltammetry data, but to make it simple, what we do is see dopamine being restored at therapeutic levels. So once we use a well-described animal model for preclinical studies that results in the loss of dopamine in the striatum, once these TENSPs, or these tissue engineered nigrostriatal pathways, are implanted into the rodent, we do see therapeutic levels of dopamine that you would expect in a non-lesioned animal. That's the graph here on the left. And on the right-hand side, there is a very well-used animal model for studying the effects and therapeutics on Parkinson's disease. And what we do see is implantation of our tissue engineered pathway does restore the behavioral deficits in a well-described animal model of Parkinson's disease. And some of the near-term inflection points, again, the ask of 2 to 3 million to allow us to complete studies that would be IND enabling and allow for regulatory engagement in the at the latter half of next year. The first of those is really expanding our behavioral preclinical studies. So I've shown you some preclinical data at an earlier time point. We would like to use this well-described animal model that really looks at the motor symptoms of Parkinson's disease and take that out to a longer time point. We would like to also assess the non-motor symptoms of Parkinson's disease. So Parkinson's disease has really been described as a motor disorder, but there's also prominent neuropsychiatric symptoms that are also treated pharmacologically. But we do believe that restoring the circuitry may have an impact on the behavioral symptoms as well as the motor symptoms. So we're looking to add some assessments of behavior that are non-motor to see the effect of our therapeutics on the non-motor aspects of Parkinson's disease. We'd like to also initiate IND enabling safety studies. So while we can't engage in the definitive safety studies until we have a final cell source procured, what we can do is begin essentially looking at the construct itself in several different safety testing paradigms. And we would like to do some safety testing again at the CRO. And it's the combination of 1 and 2 that we'd really like to take to the FDA in the latter half of next year, hopes of getting some feedback and again, going out and having a strong Series A in 2022. The combination of the preclinical studies and our early safety studies allow us also to optimize our manufacturing plan and to work to reduce the cost of goods sold and to work on developing commercial grade, clinical grade, and commercial grade material. Again, here's our roadmap. So we're looking for a pre-sub meeting in the latter half of next year, in a Series A early in 2022, and the data and the funds that we're requesting and the activities that we'd like to perform and the efficacy and the safety studies we'd like to perform from now until November, hopefully having a good readout of the preclinical efficacy studies by Q3. So I really discussed a lot about our main application, which is Parkinson's disease, our lead indication, but there are a number of other indications, again, within the central nervous system that our technology would be well poised to address, particularly disturbances in the auditory and the visual pathways, as well as looking to regulate existing circuitry rather than replace lost circuitry, where we do see aberrant neuronal firing. And two examples of that would be epilepsy and obsessive compulsive disorder. And we do also have some proof of concept data that we're aggregating for our non-Parkinson's indication. So these activities are ongoing as well. And our team, again, is Dr. Cullen and Dr. Smith as the founders, myself and Dr. Harris, who is a alumni of UPenn, are the lead consultants. We also have a great board of directors comprised of the IP group, as well as folks within UPenn. So with that, I will open up to questions, and thank you for your time.
Thank you so much, Nader. It was a great pitch. And we will move into questions. If anybody has questions, please add them. How does Intervace's approach compare contrast to Blue Rock Therapeutics acquired by Bayer?
Great, that's a great question. So Blue Rock is actually one of the studies listed here. This is the embryonic stem cells. So Blue Rock is looking to deposit stem cells within the striatum and re-establish dopamine within the striatum, but it doesn't recreate the circuitry. And this goes back to some of the very early studies that where cells were deposited into the striatum. It creates a signal, but that signal is unregulated. So what we've seen previously in clinical studies is uncontrolled dopamine results in a lot of behavioral abnormalities. So our technology is, we really think, an evergreen. So we've actually thought about a potential partnership with Bayer could be a great opportunity as a next generation technology. If we do see some positive safety data and some positive efficacy data, that doesn't prevent Intervace from operating. It's actually a follow-on next generation approach, which will be even more targeted and hopefully have less side effects than Blue Rock and Bayer's approach. Great.
Can you comment more on the current and intended cell source?
So our current cell source that we're using is a commercially available non-GMP cell line. We've done some work with iPSC and embryonic stem cells, and we really do see that both cell lines tend to work quite well within our construct. So we're looking to finalize a cell source based on a growth matrix that we're developing to see what can reduce the cost of goods sold. But we're not seeing a major effect on cell source at this point. So if that trend holds, which we hope that it does, we'll really pursue the cell source that we can acquire and partner with a CRO so that we are not a cell manufacturing company, but a tissue manufacturing company. So some of this will fall back to economics if we don't see a biological difference, and we haven't seen a big biological difference to date. So we're flexible in that.
Excellent. The question up there, I think, has already been answered. Any other questions the audience has? All right. Well, thank you very much, Nader. Excellent pitch. We appreciate you being here. And again, if anybody has more questions, they can reach out to Nader directly, and anyone else on the PCI Ventures team will get those questions over to Intervace.
Thank you.
Thank you. Next up will be Vetagenics, and we will have Adrienne Sachs, the CEO of Vetagenics, will be presenting. We will give her a minute or two to get up and coming. She can share her screen.
Okay.
I can see it. Adrienne, we'll just hold you up for 1 minute to make sure that anyone who's coming on just to see you, we don't start too early for them. You'll do it 3:15. So everybody's been doing an excellent job managing their time on the pitches to keep us right on schedule.
I see that. That is something. Congratulations to your team, Michael, for preparing us.
As somebody on my team said earlier, it's practice, practice, practice. So we went through this with everybody to make sure. Yeah. Okay, with that, we will be ready for the next pitch with Vetagenics. Adrienne, go ahead and take us away.
Okay, great. So thank you to PCI for giving Vetagenics the opportunity to present the company today. I'd like to start off by asking the audience, how many of you have seen TV commercials recently for Dupixent or Yervoy, Opdivo or Keytruda? Then I know you can't raise your hands, but assuming that some of you are, then you're aware of the transformational impact immunotherapy has had on the treatment of human cancers and other chronic conditions. In fact, the worldwide market for human immunotherapies will reach $163 billion in 2020, and it's expected to grow over 11% to $275 billion by 2025. Despite the overwhelming success of these drugs in treating human disease, less than 3 have been approved worldwide to treat pet dogs. At Vetagenics, we believe dogs deserve to benefit from this new form of therapy and realize the same therapeutic advantages to enhance and extend their lives. Our approach directs the dog's own immune system to precisely attack the specific disease without crippling adverse events. While alternative methods to generate canine-specific immunotherapies use different techniques to try to mimic the dog's natural immune response, Vetagenics' proprietary phage display platform uses the RNA from actual dogs to create precise, entirely canine, highly versatile antibody fragments or cannibodies that are rapidly isolated and designed to be non-immunogenic. Our cannibodies can then be engineered into various therapeutic constructs, including full-length monoclonal antibodies, bispecific T-cell engagers, and CAR-T for easy cost-effective administration in a veterinarian's office. To take the next step, Vetagenics needs $1 million to demonstrate proof of concept in dogs with cancer of our 2 most advanced compounds. Cancer is the leading cause of death in pet dogs. Over 50% of dogs aged 10 years or older and 23% of dogs of any age will die every year from cancer. Current treatments include surgery, radiation, and systemic chemotherapy, which ranges between $5,000 and $11,000 per treatment course. Based on market research, over 370,000 pet owners have made the choice to pursue aggressive systemic chemotherapy, which today is not tumor specific and causes significant adverse effects that limit its efficacy in treating the disease and causes devastating adverse events to the dog. Vetagenics VGS-001 and 002 aim to replace chemotherapy, offering precise, efficacious, tolerable therapies that can be easily administered by subcutaneous administration. So what are the requirements for a successful canine immunotherapy? Well, it has to be fully canine to avoid rejection or neutralization by the dog's immune system. It has to be highly specific to the canine antigen with subnanomolar binding affinity. The candidates also need to have high developability to ensure cost-effective production and ultimately a low cost of goods. And the candidates need to demonstrate functionality and developability early in the development process to quickly de-risk candidates as they advance through development and increase cost of development, which we'll comment on in a moment. So Vetagenics solution, our cannibodies offer an answer to these developmental challenges. They're unique, fully canine antibody fragments to any canine antigen that are uniquely isolated from our proprietary phage display libraries and further selected by Vetagenics' innovative panning processes for specific desired traits. Once selected, cannabodies are ready to be immediately formatted into the therapeutic modality of choice, be it monoclonal antibody, BiTEs, or CAR T. Additionally, these therapeutic formats do not require any time-consuming canonization or affinity maturation. Cannabodies are also not dependent on pre-existing murine or human antibodies and require no animal pre-immunizations or sacrifice. Cannabodies are rapidly generated and developed and can go from target identification to lead candidate in anywhere from 3 to 6 months. This chart provides a look at Metagenics cannabodies versus competitive technologies. Some of the points here I've already mentioned. You can see on the far left the technologies that are currently being used to generate canine immunotherapies. But when we look to generate these antibodies, what is it that is the priority? The priority is to generate entirely canine antibodies to minimize or eliminate immunogenicity. We want the ability to select our cannabodies or our antibodies from the full antibody repertoire of the actual canine immune system to make sure that we are not limiting the potential combinations that could be an excellent fit to target a specific antigen. There are no mouse or other pre-existing mAbs required, and the process is rapid, productive, and cost-effective to get to a lead candidate. So for those of you that are not familiar with the animal health market, animal health represents a significant opportunity as more people than ever are adopting dogs and other pets as an important part of the family to provide comfort and stability, especially in these uncertain times. And they're willing to pay out of pocket for their care. As you can see from the top graphic, therapeutics to treat animal diseases require a shorter and less expensive development path. And although these drugs don't generate the billion-dollar revenues of their human counterparts, they offer sustainable cash flow, and they are not subject to generic competition or third-party payers. As you can see in this graphic, cannabody development is a very streamlined process, offering a strong return on investment with de-risking pilot trials early in development. The larger investments required for commercial production and registration have a higher probability of success and can produce revenues within 2 to 3 years from that investment. What's great about cannabodies is they're very easily and quickly developed. And you can see from the time of lead candidate generation, we can be generating revenue within 4 years. Our team of highly experienced scientific and business leaders from both human, biotech, and animal health are focused on guiding the development of this exciting portfolio of therapies towards a new standard of care in treating animal cancers and chronic disease. We further benefit from an extensive network of industry experts to round out all key aspects of the business, including technical, legal, IP, and regulatory. As you can see here, the investor and partner interest is growing in companion animal immunotherapies, recognizing the demand from pet owners who want better, safer, life-saving treatments and are willing to pay for them. Metagenics is excited to share that we are currently in advanced discussions with 2 large animal health pharma, which speaks to the importance and novelty of potential vet biologics for the future. So in summary, Vetogenics is devoted to saving lives of companion animals, driven by transformative therapies. And we're also proud to say we're located in the heart of Silicon Valley, Philadelphia, a resource for growth and innovation. We have a novel phage display platform technology that allows us to generate fully canine, highly specific drug candidates that are designed to be non-immunogenic. There are significant opportunities beyond cancer for other areas of unmet medical need in animal health: allergy, pain, inflammatory disease, infectious disease. Also, the company has grants and is pursuing third-party partnerships to maximize capital efficiency. And finally, we are seeking $1 million to generate proof of concept in our 2 lead cancer compounds, to identify some additional opportunities in other areas of high unmet medical need, to further our product IP for our 2 lead compounds, and to strengthen our infrastructure to support our growing and advancing pipeline. Thank you for the opportunity, and I'm happy to take any questions.
Thanks so much, Adrian. It was a great pitch. And now we'll open it up to questions. So the first question was, have you thought about what the equity terms would be on a deal?
We are looking at a variety of different opportunities, both equity and debt. And we are, I think right now, we just started our capital raise, so it's a little premature. But from a debt standpoint, because that is the conversations that we've had, we are looking at a convertible debt that would come with warrants and an interest rate of about 7% and a 20% discount upon the conversion.
Excellent.
But we are very open. We are very open to equity and would entertain those discussions and considerations.
Great. Any other questions from the audience? So what percent of the market potential do you think that you can capture?
So initially, we believe that we could capture 10%, which is fairly significant if you think about $10,000 per course of therapy. But the way that we're looking at this right now is that there are about a little over 400 veterinary oncologists that are treating that 370,000 dogs, because they're requiring systemic chemotherapy, which cannot be given in a community veterinary office. So even though we look to capture about 10% at the outset, one of the opportunities we see here with our products is that because they don't require special handling, they're not given through intravenous administration, that they can be used in a doctor's office. And so we do anticipate that the market will grow. And so these compounds, we're looking anywhere from 50 to 150 million per drug, but we do hope that will expand over time.
Excellent. And could you talk a little bit more about your IP portfolio?
So currently we have an exclusive license from Penn for the use of the canine fully display phage display libraries for veterinary use. We are also in discussions to license VGS-001. The patents are just being filed, so we're going to also be getting the exclusive license for VGS-001, which was originally initiated at Penn, so that will become exclusive to the company as well.
Excellent. Well, that's great. I think we'll close it off there. Thank you so much, Adrian. Excellent pitch. And we will be moving now onward into our advanced materials section. We have next up will be Hydropore. And I also wanted to mention to everyone, please follow PCI Ventures on Twitter, LinkedIn, and Facebook so you can continue to get portfolio updates as they move along. And now we will move into Hydropore with Peter Lorenzi. Peter, I can see your screen.
Great stuff. Can you hear me as well, Michael?
I can, and so I'll hand it off to you, Peter. Take it from here.
Good, good. And you can see my screen in presentation mode. I just want to check.
Waiting. It's still showing it. Yeah, try that again.
There you go.
There it is now. You're good. Take off.
Okay, thank you, Michael. So Hydropore. Hydropore makes hydrogen fuel pellets for drones, and we have two major benefits. The first is that we can increase the commercial drone pilot productivity by up to 40%. And the way that we can do that is by eliminating the need for lithium-ion batteries and all that goes along with that in terms of charging and swapping out batteries. And the second benefit is that a Hydropore fueled drone can fly 3 times further than a regular battery-powered drone. And we think that's going to open up entirely new applications for drones. You may not know this, but Amazon Prime Air was in August was granted approval by the FAA, the Federal Aviation Authority, to use its battery-powered drones for delivery. And of course, Amazon makes about 400 million deliveries a month. Delivery is one of its biggest costs. And we think that Amazon and others like Alphabet's Wing will be quite interested in a drone that can fly 3 times further. But we don't intend to call Amazon just yet. In fact, we're going to start off with our target market of mid to small size US agricultural drones. And we're targeting them because flight time for them is a real issue. They fly 500 to 1,000 acre fields, and they are in desperate need of something that can go further. They're also willing to try out new technologies because they fly out in the open. And we estimate that the total accessible market for Hydropore in that sector is somewhere in the region of $600 million. Our product is single-use Hydropore fuel pellets. They're aluminum, they're made out of nanoporous aluminum. You add water to them and they produce hydrogen gas. And at the moment, we're assuming our average selling price would be somewhere in the region of $40, which will give you about a kilogram of pellets and about an hour's worth of flight. And the technology works. We've demonstrated that we can power a car in the lab, and we're currently building pilots and prototypes to show that it can fly in a drone. And we're seeking $450,000 in pre-seed funding to do 2 things. One is to complete those prototypes, and secondly, to prove that Hydropore fuel can beat lithium-ion in terms of flight time. So I talked a little bit about batteries. The short flight time really constrains what drones can do, and it creates a lot of downtime. A pilot has to monitor how far a drone has flied, and monitor the batteries, bring it back, swap out the batteries, fly it back to the flight pattern and continue on. And of course, there's a lot of charging that occurs. And the 30-minute flight time is really only for new batteries. As they cycle through charging cycles, that performance diminishes. And actually, in cold weather, the performance diminishes even further. So Hydropore has really solved this by making a fuel that can deliver 90 to 120 minutes of flight time. Of course, without all that downtime of swapping batteries, the drone pilot can be 40% more productive. We're not requiring the drone operator to buy a new drone. In fact, you can just swap out the hydropore fuel system in place of the batteries. And of course, we are not constrained by cold weather either. So here's a use case, and this is actually a real use case. We're doing some customer discovery with agricultural companies in the Midwest. And Sean here, who works for ACA, calls us up wanting to buy some hydropore fuel. ACA does trials for agricultural companies like Bayer. They own 20 large drones, and they subcontract to commercial drone pilots. And basically, the moment they can have those pilots fly one 500-acre field a day, and with hydropore, they can fly 2 fields a day. And those kind of productivity savings will yield about $400 in savings per day for ACA, and that's pilot time, and that's after the cost of the hydropore fuel. So as I said, we estimate the total accessible market in the ag sector is $600 million. I'm happy to talk through some of the assumptions on this in the Q&A if you have any. So I've talked about lithium-ion batteries in terms of the competition, and lithium batteries are really the dominant fuel and power source for multi-rotor drones. They're not the only options. Gasoline drones also exist. They're very rare, and they're rare because they have real reliability issues. And because of that, there's a lot of maintenance, a lot of downtime. And they're also very noisy, and they vibrate, which affects the instrumentation on board. Hydrogen drones also exist, and they carry hydrogen in tanks. And the big issue there is that dealing with those tanks is really expensive and complicated. You need somebody, a specialist, a supplier, to bring hydrogen to your takeoff site. You have to use a special compressor to bring it up to 6,000 PSI. And that just makes it far too expensive and far too unwieldy, despite the additional flight time. So hydropore, we feel, gives that sort of leading flight time, that leading performance, and is actually easier to use than lithium-ion batteries. So just a quick word about the team. The 2 founders of hydropore are Eric Detsy and John Corsi. Eric is the Stevenson Term Chair Professor of Material Science and Engineering at Penn. He's got a really exciting and up-and-coming lab that's attracting a lot of funding in materials and batteries. And John is a PhD student in Eric's lab. And actually, it's John's work that forms the underpinning of hydropore. For myself, I have 15 years experience in R&D, in pharma R&D, in fact, in industrializing platform technologies. And I also have a physics background like Eric and John. And we have some great advisors and board members. Michael, of course, as a board member, Ruby Shandy, who has been a senior marketing executive in public companies and is also a board member of public companies, and Mark Doyle, who's the former CEO of DuPont. In terms of the risks, we're early, and there are obviously things that we still have not yet evaluated. The most significant of those, I would say, is the economic viability of the manufacturing process at commercial scale. We've already scaled up 1,000 times in the lab. The raw materials are inexpensive, but we just haven't assessed the economics of that yet. And we also don't know whether we are going to get approval, whether there are going to be restrictions on transporting hydropore fuel. You may know, of course, that lithium-ion batteries cannot be carried in the cargo of airplanes, for example. So that's it. Just in summary, hydropore delivers 40% of more productivity for drone pilots, which we think is incredibly compelling in the commercial drone sector, and 3 times longer flights, which we think companies like Amazon and Alphabet's Wing will be very interested. That's it. I'm happy to take any questions, Michael.
Excellent. Thank you, Peter. Great pitch. Let's see. So of the total addressable market, what do you think that you can capture?
That's a good question. What we, in our assumption on the total addressable market, we assumed that we could capture half of the flights. So there's somewhere in the region of 30 million flights, and we feel that we could potentially capture half of those. Agricultural drones are big drones, and they need that extra range, and they're continuously flying big fields. So we've already taken the total market, and we've assumed a 50% sort of attrition on that, and so 600 million is where we're coming in at.
Okay. Can you talk a little bit more about your IP portfolio?
Yep, absolutely. So the patent is filed and pending for the process for making nanoporous aluminum, which is the active ingredient of hydropore fuel. And we have an exclusive option from Penn to license that technology.
Mm-hmm. Can you talk a little bit about the dangers of hydrogen and how that affects your product?
Yeah, I mean, hydrogen is an explosive gas, and one of the things that's good about our product, if you compare it to hydrogen gas in tanks, for example, is that we don't need to operate under pressure. We produce hydrogen on demand when you need it, rather than having to transport it around. And actually, as a gas, although you have to be very careful, sort of, if it leaks inside, outside it escapes up into the air very, very quickly. And so, you know, the dangers of hydrogen, you have to be careful, but we don't think that's going to be a significant, issue for us.
And you're confident in the stability of your pellets as well?
Yeah, we are. And in fact, one of the things that one of the additional things is that we mix the nanoporous aluminum, which is really the active ingredient that makes the hydrogen. We mix that with other ingredients and form it into a pellet. And we're able to kind of fine tune the attributes of those pellets to make it either more stable or to react quicker. And that ability to kind of fine tune the pellets and adjust the recipe, if you like, of the pellets, we think might be actually another place where we could get IP coverage as well.
And have you thought about the terms of your $450,000 raise?
At the moment, we haven't. We are at very early stages, so we're open to conversations.
Excellent. Any other questions by the audience? Okay, if that's it, if anybody has further questions, feel free to reach out to Peter. It was a great pitch. Oh, wait, there might be one more. Have you achieved revenues yet?
We have not.
From that. Okay, great. Well, thank you so much, Peter. I appreciate it.
Thank you for your time.
Absolutely. And next up, we have Enichip with Trifon. I am going to avoid saying his last name because I will get it wrong. But Trifon and I have worked together for quite some time. We're happy to have him here today, and he will be pitching Enichip. And Trifon, your speaker is on mute. You can unmute.
Hi, yes, thank you, Mike.
Great, and you've now got a minute or two to get your screen share up.
Okay, setting should come up.
There we go. I can see your screen. If you want to go into presentation mode.
Yes, I'm going now.
Excellent.
It's better?
With that, we will hand it off to Trifon for his presentation on Anychip. Thank you so much. Take it away.
Thank you, Michael, and thanks everybody who sticks around to the end of a long day to listen to Anychip. So my name is Trifon Liakopoulos, and I'm a CEO and co-founder of Anychip, a startup company that addresses the root cause of a power integrated power management inefficiencies in today's electronics, and with a key game-changing innovation and breakthrough technology. This technology platform impacts energy savings and cost and size reduction, which in return enables vertical power integration across multiple industry verticals. There is a significant experience and recognition on the the company's leadership team, and that drove an unusual early traction and strategic engagements with tier one world-leading companies. So what is the root cause and the problem? The problem is in today's electronics, the power is separate from the active silicon chips, such as the CPUs, GPUs, and artificial intelligence processors. As you can see on this latest double redundancy Tesla computer, where each of these processors is getting powered by a series of distributed microchips and circuits. And actually, every of these processors has about 1,600 input-output connections on the backside. And most of them is about power delivery and transfer. So if we can imagine today's power transfer from the source to where the demand is distributed across a board, as a traffic network, there are a lot of inefficiencies. There are wasted cycles waiting for power to arrive, there's congestion, there is a fixed size delivery of the energy transfer, which is usually a magnetic passive component, like an inductor and transformer, signal complexity, etc. So if we imagine that we can reduce the delivery vehicle, the magnetic component, and make it smaller, we can transfer energy much faster, and we can also minimize the routing. So basically, we can bring the power source right underneath the microchip or the processor and drive the power where the silicon is needed. So we can integrate the silicon and the power together in a vertical power delivery and activate and provide power where it's needed, when it's needed, and even in more sophisticated software enhanced implementations, we can predict where the power is needed and deliver it. So why this thing is not happened so far? It's about the size of the magnetic devices and the size of these components. Well, as you can see here is the largest component on the board. And on top of that, if we try to miniaturize these components, then we have a performance collapse due to dramatic size of the magnetic cores and materials who cannot operate at high frequencies who allow this reduction. So Anychip breakthrough innovation in power offers the only commercial viable technology so far that integrates the power on silicon chips. So it takes this big passive component and brings it right on top of the silicon of the IC, and we can create what we can call monolithic power delivery. We go from power system in a package to power system on a chip that is very desirable. How we can achieve that is, and what's the differentiation of Anychip? We have a very high frequency specially designed magnetic alloys who can basically take a three-dimensional bulk magnetic component and make it a 2D, put it as a thin film on top of silicon. In order to do that, you need to have a much higher performance and efficiency alloys to do that, and materials, and that's what we have. We also have a game-changing manufacturing processes that are breaking the existing power integration cost barriers. In order to achieve this kind of devices, you need something like a multi-layer process who requires a lot of number of steps and masks, fabrication process complexity that is so far it's cost prohibitive, and that's a big barrier. We have simplified this process dramatically, going producing the same results with only 5 steps and 1 mask. That gives a significant cost reduction. And the key here is that we are using the existing equipment and processes that are available to today's microfabrication and IC fabs. We have a quite strong patent portfolio. We have exclusive licenses from UPenn. And we also have applied for multiple patents, and then we continue applying for patents that have to do with the material composition and manufacturing technology and even designs. So the invention of the transistor at Bell Labs about many years ago changed the electronics technology and lead to ultimate circuit integration. So same way I envision today NXIP's technology platform will lead the integrated vertical power revolution to similar breakthroughs. So we take a big component similar way and make it into integrated silicon mode, and that allows the integration. I'm very keen to this analogy since when I started my career back at Bell Labs, I worked basically on the same lab. It was called Slimline at the time that the transistor was invented. I noticed that later from a plaque on the wall. So NXIP's fundamental process modules are having the roots on key innovation. So we have, as I mentioned, a good current VC support. We have seed funding from institutional investors who also have committed to participate on the next round. We're seeking to raise $50 million in Series A. And we also have quite, as I mentioned, strong corporate partnerships. We have engaged with the world's largest #1 component manufacturer, which already came in as a strategic investor, pre-Series A investor. And we also have engaged with the world's #1 analog product manufacturer to develop a device that will enhance and reduce the cost on existing product line that they have in signal processing. So we also got funded as part of a European consortium, the only non-European company for a medical device using a micromagnetic actuator. So we have a process, a technology process, and enabling technology platform who can be applied to different applications. So how we plan to go to market? The idea here is to basically, with the Series A funding, we want to commercialize the phase 1 towards phase 2. Phase 1, we work directly as we do today with a customer to develop the components and the process modules that are necessary for this integration. Out of this, at the same time, we already discussing for where to transfer and license this technology with leading foundries across the world. And we are creating libraries. And these libraries basically will allow the customer to design their products. And we're looking on a license model. On phase 2, we're going to step out of the way. We're a small company and we don't want to be a bottleneck for high volume scalability. And we are going to have the customer using NXIP's library, working with the foundries directly, and then create their product. So basically, it's almost an ARM revenue model where the customer will be licensing the libraries. And at the same time, also, we're envisioning getting material use license from the foundry as well. So it's we are not having an envision to go to high volume manufacturing, but we want to capitalize on the enabling technology for different applications. As I mentioned, the team is, I'm lucky to be working with very experienced people in the industry. We have Matt Wilkowski in the line also as our VP of technology with over 40 years of experience. And the other co-founder of the company is Mark Allen, who is a professor at UPenn. And he's a leading figure in integrated magnetics, in micromagnetics. And among all ourselves, we have about 100 years of experience, about 90 patents, and several startups. Matt and I was in a previous startup where we basically commercialized the first concept of integrated magnetics. And this company was successfully exited, acquired by Altera, and then Altera was acquired by Intel. So my last slide is basically summarizes the advantages, the disruptive energy value with reduced power consumption, smaller chip sizes, significant cost manufacturing reduction, and addressing with the platform many markets towards vertical power integration. And even we can have software specific developing, hardware specific software development where we'll allow power savings even at a high level with applications. So I'll stop here and I'll take any questions.
Excellent. Thank you, Trifon. Great pitch. Appreciate that. We'll go into the questions. So just to clarify for the audience, who is your end customer?
So this is all the big corporations who manufacture, design, manufacture power management ICs, companies like Texas Instruments, Analog Devices, Infineon, Dialog, all these big players. Also the passive component manufacturing companies that can basically license or sell just magnetic components to this first group of people.
Mm-hmm. And in terms of the round, do you have a lead investor identified and any idea on valuation?
So we are looking for a lead investor. As I mentioned, our current investors have committed to participate. I'm looking to have a consortium of institutional VCs and strategic investors who are already, we have one, and I would like to bring at least one more strategic investor. So the valuation, it's for now, we're looking something about 20-25 million.
And I'm not sure if you went over it, but can you talk a little bit more about the uses of the funding, where it's going to go to?
Yes, so basically we want to create, it was one of my previous slides, we want to create, to enable this kind of engagement. We need to basically establish ourselves as kind of transfer the technology to the foundry and make it available as part of a library system. And when I say library, I mean process development kits and design kits that the customers will take. So we want to kind of go from a small lab that we have today to a more enhanced prototyping capability, so we can do a seamless transfer of the technology to the foundry.
Excellent. Let's see. Have you achieved revenues yet?
No, other than the NREs that we have engaging with this customer. So we have brought about 600K total on kind of this research project, but not revenue from selling anything right now. Just R&D and non-recurring engineering expenses.
Would you be open to tranching the round or doing a smaller round first and larger round later?
Yes, we just, we're very flexible at this point. It's just going to be able to, probably discuss if it makes sense to have a tranche round. Yes.
Okay. Any other questions from the audience? I think that's it. Trifon, thank you so much. Excellent pitch.
Thank you.
And with that being our last company, I want to wrap up for everyone. Thank you so much for staying through all of the companies. It's been a great audience today, great questions from the audience, which I really appreciate. And all the teams stayed right on time, which I think is absolutely amazing that we wanted to finish at 4 o'clock and it is 3:58. Nice job, everybody involved. Again, I want to thank my team. Fantastic job, guys. We really handled this well today. And please, if anyone has any questions or wants more information, feel free to get back to any of the companies directly or to us on PCI Ventures team. And as we pointed out earlier, feel free to follow us on LinkedIn, Twitter, Facebook. Along the side, you can see our handles there. And thank you so much, everyone. We look forward to doing this again soon. Hopefully everyone's found it to be productive. Thank you so much.